So, Dr. Prachi is joining. She's facing some problem. She will wait for another 1 minute, then we'll start, sir. Yeah, sorry for the inconvenience that Dr. Prachi is facing some trouble joining. Hopefully, we wait for another 1 minute and then we'll start.
Dr. Gopinath, the whole tried to join and she is saying that both again removed her.
Just check it out.
I'm sending you the screenshot.
Maybe we can change the order and start.
Yes, sir.
To join another event at 9 a.m. So,
Okay, sir. Okay, sir.
yes.
Yes, sorry for the inconvenience and delay. Sir, maybe you can mute your mic. It is giving echo. So, very good morning to all. It's my pleasure to welcome you all for this Indo-Canada one-day workshop on Nano-Bioengineering. So, we are honored to have Professor Ajit Chaturvedi, the Director of IIT Roorkee, and Dr. Prachi Kaul, the Director of Shastri Indo-Canadian Institute, as chief guest for this workshop. We also honored to have Professor Armugam, Dean International Relations, and Professor Pravindra Kumar, Head Department of Biotechnology, IIT Roorkee, to grace this inaugural session. So this workshop aimed at being a forum to gain insight into the recent development in the field of nano-bioengineering. It is a unique opportunity for the participants to update their knowledge in this emerging field, which could motivate new discoveries that would transform the field. With this brief introduction, now I would request Professor Ajit Chaturvedi, the director of IIT Roorkee, to say a few words about our institute and start the inaugural program. Sir.
Thank you, Professor Gopinath. And Dr. Prachi Kaul, I guess, is still not joined. So Dr. Prachi Kaul, in any case, as the director of the Shastri Indo-Canadian Institute. Then Professor Devika Chitrani and Professor Stefani and Professor P. Armugam, Dean of International Relations, Professor Pravinder, and I can see Professor Shiva Kumar has also joined this early morning inaugural, and all those participants who are listening to this inaugural function and this workshop. I think it's a very great event where Shastri Indo-Canadian Institute has brought two universities together, IIT Roorkee on the Indian side and University of Victoria on the side of Canada. And these two great institutions have joined hands to deliver this workshop, in which I can see speakers from other institutes have also joined, like Professor Shri Shiva Kumar and Professor D.S. Katti have joined from IIT Kanpur. I was wondering if it was not Corona time, then this workshop may have been possible physically, and maybe we could have an opportunity to meet and exchange notes in addition to the workshop. But while that pleasure is still a few months away, it is very heartening to see that because this event is online, it has attracted a very huge number of participants. The number of registered participants, I'm told, is now more than 1100. This would not have been possible if we had a physical event. So there are pros and cons. While it would have been better if we had it physically, but then we would not have got the kind of audience that we are getting today. So this kind of events, I think, are very important because the topic of today's workshop is of interest to a lot of people. I mean, whether you are doing bio, whether you are doing engineering, both parties, and in particular, if you are doing anything in the nano domain, and a great fraction of academicians are now working on nanoscience, nanotechnology, nanoengineering. So this kind of workshop will be very timely for them. Students, even researchers, established researchers who want to pick up new dimensions, new areas, they will find something very useful from the 5 talks which have been lined up on 5 distinct topics. And the good thing is that each of these talks is not like a very preliminary talk. It is talking about deep research and for a long period of time. So I'm sure that these talks will have a very good impact, not only on IIT Roorkee students and faculty or University of Victoria students and faculty, but also to all the other audience who have joined globally. I wish the participants go away with something useful takeaways, and I also hope that the speakers get something useful out of this workshop. I wish this workshop a great success. Thank you, Professor Gopinath.
Thank you very much for your kind words. Now I would request Professor Arumugam, Dean International Relations, to say a few words about our International Relations Office.
Thank you, Professor Gopinath. Good morning to everyone in India and good evening to the others on the other side of the globe. In fact, it is a nice meeting. Like Professor Chaturvedi said, it's possible due to the SICI. And SICI has been playing a very crucial role in building collaborations between many Indian institutes and Canadian institutes. This has gone a long way. In fact, even in our own institute, the collaborations have increased many folds. This event, as a part of that SICI initiated collaboration, seems to be a very good success. And of course, our technology center and the biotech department, it's a very rapidly growing department, I should say. I could see that there is a nice interaction between Professor Gopinath and the other professors like Professor Stefani and Professor Devika Chitrani. So I certainly would like to invite you here. Like Professor Chaturvedi said, you couldn't come during this event, but certainly in near future you should come here and spend more time with us. we will be happy to host you. Even our institute has support to host foreign faculty beyond what is already available through SICI. So this also certainly you can think about. And there are several opportunities which we can initiate. For example, with University of Alberta, Canada, we have a joint PhD program, and I'm sure that you can identify some students in this meeting and some professors in this meeting with whom you can interact and have a plan for jointly supervising some PhD students and so on. So these joint degree programs, our office certainly will facilitate and take care of the bureaucracies you don't have to worry about. And of course, to be particular to University of Victoria, Canada, I can see that our institute has a good collaboration with University of Canada. Starting from 2007, I can see the joint publications. So we have interactions with your mathematics faculty, electronics, and hydrology, water resources, and so on. So there is a good background between our two institutes. I hope this will increase in the near future. As I already said, you are all welcome to our campus. We hope this collaboration will lead to sustainable and long-term research network. Thank you very much.
Thank you, Professor Armugam. In addition to that, Professor Shastri Indo from IIT Kanpur, he is also from the University of Victoria. So that is the one added. One of the speakers also we have from the same university. And now I would request Professor Pravindra Kumar to say a few words about our Department of Biotechnology, IIT Roorkee.
Good morning, all honorable dignitaries today in this one-day workshop on nano-bioengineering. And I welcome all of you from on behalf of me and our department. So our department was started long back in 1980, which is evolving and rapidly increasing also. So the department focus is basically to provide the academic training as well as to do cutting edge research in interdisciplinary areas. So it's really great that our department, we have energetic faculty members. Most of them are dynamic, young. You can see Professor Gopinath, who's been publishing, doing a lot of patents. So most of us are really involved in both teaching as well as in doing research in interdisciplinary area. Particularly, we emphasis on health sciences. And our department consists of different areas we have made it. One is computational and structural biology. Other one is cell and molecular biology. The third one is biological engineering. So we are focusing in 3 areas, and we have different academic programs. For example, we have BTech program in biotechnology. We have MSc in biotechnology, MTech in bioprocess engineering, and PhD. And all the students is about 600 in our department. We have about 25 faculty members, and this department has been doing good work in all the fronts. And today I don't want to take much time. I welcome all the eminent speakers and participants to this event, and I say all the best to all of you. Thank you. Thank you very much, Professor Gopinath.
Thank you, Professor Pravindra. And due to some technical glitch, Dr. Prachi Gaul, she was not able to join. So she may be able to join in the validatory session. Okay. So right now I'll request Professor Devika to say vote of thanks.
Thank you, Dr. Gopinath. Let me first extend my thanks to the Honorable Director of IIT Roorkee, Professor Ajit Chaturvedi, and Dr. Prachi Kahul, Director of Shastri Indo-Canadian Institute. I'm sure she will join us later. I hope so. And I also would like to extend my thanks to Dean International Relations, Professor Armugam, and also the head of the Department of Biotechnology, Biotechnology, IIT, that is Professor Ravindra Kumar, for spending their valuable time with us to grace this occasion on Saturday morning. And we would also like to extend thanks to Shastri Indo-Canadian Institute for sponsoring this workshop, and special thanks are also due to all the staff and students who made the arrangements for this workshop. Once again, I thank you all for joining this inaugural session. And now I would request Dr. Gopinath to introduce the first speaker of this workshop.
Thank you, Dr. Devika, and thank you all the dignitaries who joined the inaugural session. So now we can conclude the inaugural session and we can move on to the first talk by Professor Stephanie Villiers. So Professor Stephanie Villiers is a full professor in biomedical engineering, holds the Canada Research Chair in biomedical engineering at the University of Victoria. So she has dual appointments in the Department of Mechanical Engineering and the Division of Medical Science. She also holds an appointment with the School of Biomedical Engineering at the University of British Columbia. So she serves as an acting director at the Center for Biomedical Research and the Biomedical Engineering undergraduate program at the University of Victoria. She is an active member of the steering committee of BC Regenerative Medicine Initiative and the Stem Cell Network. She also serves as a staff scientist at the Creative Destruction Lab. She was also the president of the Canadian Biomaterials Society. She has recently founded a startup company, Oxetol Biosciences, and she has a huge number of publications and patents. With this brief introduction, now I would request Professor Stephanie Villiers to deliver her lecture.
Thanks so much. That was a wonderful introduction, and I'm so excited to be here virtually to share some of the work I've been doing, and thanks again to all the organizers and the sponsors. Hopefully, okay, I think my screen should be sharing now. Yeah, that was a wonderful introduction, and as many people have mentioned, it would be really lovely to do this in person, and hopefully we're getting there now that vaccines have been rolling out. But you know, once travel is possible, we are always happy to host students in our lab at the University of Victoria, which you can see right here. Our campus is inside of a ring road, and my lab in particular is located at our medical school on the campus. And one of the things that we've been really lucky in my lab is to we have access to a large number of bioprinters. And so that's what I'll be talking about today, is a lot of our 3D printing technology that we use to screen different types of drugs. So thanks everybody for coming. So just to sort of set the stage for our work, my research focuses on tissue engineering, which you apply the principles of both biology and engineering to develop substitutes for diseased and damaged tissue. And this field has been around for decades now. And there are things such as the artificial bladder you see up here, where we actually do have artificial tissues and organs being transplanted into people. So it's a real field. These are some of the classical examples. This is the Vacanti mouse, where they grew a human ear on a mouse. This was the controversial tracheal implants, which ended up being the result of some fraudulent data. And then you can see here in the corner is neural tissue engineering, which is what my lab specializes in. And one of the reasons you see often stylized photos is neural tissue engineering is quite difficult to get right as compared to some of the other tissues in the body. And this is one of my favorite slides to show, which as an engineer, it shows the spectrum of what you need when you want to make the different types of tissues. So for things like your bone and cartilage and tendon, you're really replacing a structural component of the body, and the corresponding tissues are designed accordingly. When you're looking at things like the liver or pancreas, which some of my colleagues across the water over at UBC are doing, you're really replacing a metabolic function. And then there's sort of this area in the middle where it gets a bit tricky. Things like your nerves, your muscles, especially like the cardiac, heart tissue, and skin, have both these structural components as well as these metabolic functions that both must be recreated when you're engineering them. And so that's what's going to guide your principles for making these types of tissues. I've always been intrigued by neural tissue engineering, starting with my graduate work, where I worked with Dr. Kelly Sakyam-Elbert studying how to make replacements for the damaged spinal cord. One of the things that is interesting about traumatic injuries to both the brain and spinal cord is that there aren't really any good cures. And so some of you might be familiar with racing. I know India did host an F1 race there for a while, a few years back. I'm a big fan. Possibly the greatest F1 driver of all time, Michael Schumacher, suffered one of these horrible traumatic brain injuries while skiing. And even with essentially the unlimited resources afforded to him by being one of the most highly paid athletes, they really haven't seen much progress in his treatment, just to show you how someone, even with pretty much unlimited resources, really can't recover from these types of injuries. The same goes for neurodegenerative diseases. I'm sure most of you know someone who's suffered, have grandparents or loved ones who've suffered from diseases like Alzheimer's or Parkinson's. And here in Canada, there's a really high incidence of multiple sclerosis. And again, there just aren't good long-term cures for these diseases. And so that's why it's really important to figure out how to engineer healthy neural tissue to try and treat some of these diseases and disorders. With our 3D bioprinting work, when we're printing our neural tissues, the goal is to develop tools to help us discover better drugs. And so currently right now, the drug discovery process is inefficient and extremely expensive. And in neurological conditions, approximately 90% of the drugs go to clinic fail. And so that's what we've been trying to do with our neural tissue models, is to make better ways of predicting what drugs are going to be effective. And I'm going to close out my talk by giving a really elegant example of how we've used 3D printing to make some humanized tissue models for drug screening. So one of the reasons they have such a high failure rate is, and I'm sure for a lot of the grad students in the audience, especially if you're working in biotechnology, you might already work with animal models, which are currently the gold standard. But as grad students know, they are quite labor intensive depending on the model and the treatments you have to administer. And then from the PI's perspective, they're also extremely expensive given their lack of predictive capability. You can also use human tissue slices, but they tend to come in limited quantities as it's very hard to get actual fresh human neural tissues for obvious reasons. So because of that, my lab that I started at UVic in 2010, we've really focused on using stem cells as a tool for engineering neural tissue. And I, in fact, wrote a whole book about this when I was on sabbatical at the University of Wisconsin back in 2016. And the stem cells we work with are human-induced pluripotent stem cells. And so these cells are essentially adult cells that are reprogrammed back into being a stem cell that can become any cell type found in your body, as you can sort of see here along the bottom. And so what we try to get these stem cells to become is to become the cells found in the central nervous system. And so in this case, we mainly focus on making different types of neurons, which are the excitable cells that transmit and receive information throughout our bodies. And you make different types of neurons if you're trying to replicate different regions of the brain compared to the spinal cord. There are also oligodendrocytes, which insulate the neurons by covering them with a myelin sheath, and these help them transmit their signals. And finally, there are astrocytes, and these both provide nutrients and support to your neurons and oligodendrocytes, but they also play an important role in the immune system, as well as making sure that your central nervous system, the brain and spinal cord, are immunoprivileged by making up a structure called the blood-brain barrier. And I'm not going to go into it today, but I do have colleagues such as Ethan Lipman, who's at Vanderbilt University, and Cheryl Wellington, who's at the University of British Columbia, who actually spent a lot of time developing these blood-brain barrier models, because essentially it really does, as the name suggests, pose a barrier to drug transport into the brain. And so that's another reason why we have such high failure rates when you're looking at these different drugs for treating neurological conditions, is the drugs can't even reach the cell populations they're targeting. So one of the questions that comes up is you have these stem cells that can become any cell type found in the body. So how do we get them to become these neurons, oligodendrocytes, and astrocytes? And so one of the things our lab has really used as an important tool are small molecules. And they can work in a manner similar to growth factors in terms of promoting cellular proliferation and differentiation, but they tend to be less expensive. There's tons of them out there. Lots of small molecule libraries exist. And I'll talk about some of our drug encapsulation efforts into our microspheres. And so it's nice because they're less sensitive to conditions like temperature and pH, as opposed to things like growth factors that denature. And so it's much easier to encapsulate them as well. So in our group, we've really focused on 3 different small molecules. The first one, retinoic acid, is a canonical factor that's been shown to pattern stem cells into neurons for decades now. It's a vitamin A derivative, and it really establishes polarity in your body. A retinoic acid gradient during development is what actually patterns your spinal cord. And so this is what forms your hindbrain, as well as your neural tube that becomes the spinal cord. Puremorphamine is a small molecule agonist against the protein smoothen. So this mimics the protein sonic hedgehog, which was named after the video game, which also I believe they've now made into a an animated movie as well. So that's a fun fact. And this pattern's the ventral tube, and it actually specifies motor neurons, which is a type of neuron that are lost during spinal cord injury, which I'll talk about when I talk about some of our spinal cord work. And our group back in 2015 showed that this molecule really efficiently would make neurons and motor neurons from induced pluripotent stem cells. And some work from my lab at this time also validated a small molecule called glucosterone, which is a plant steroid that's usually used as an anti-cancer drug. But a group, Gonzalez et al., they published a scientific reports paper where they actually screened 1,000 small molecules to see which ones would most effectively make dopaminergic neurons from these pluripotent stem cells. And dopaminergic neurons are the cells that are lost during Parkinson's disease. And so this was the molecule they found that really effectively promoted differentiation of these pluripotent stem cells into dopaminergic neurons. And our lab replicated that with some nice work done by Megan Robinson, who's my former lab manager, showing we could do this for human-induced pluripotent stem cells, and we actually were getting functional dopaminergic neurons out. So when I started my lab, we were doing a lot of our tissue engineering, polymerizing our scaffolds and all those things by hand. And so here are some things that I and my students observed when we were engineering neural tissue. One is that differentiation into functional neural tissue takes a long time. It takes a couple of months. And if you just culture these neural aggregate cultures, they sort of expand out in all directions. And this is actually an example of an organoid, which is another one of these types of aggregate cultures that we've made in our lab. And so you can spin them and treat them with factors, and they'll resemble mini brains. But one of the reasons pharmaceutical companies are slow to adopt them is because they have a lot of variability between the different types of organoids. And so with that, we actually really decided to pursue 3D bioprinting as a way to address this. And we were quite lucky because in Vancouver, around when I was around like 5 years into my faculty position, was when Aspect Biosystems, one of the leading edge bioprinting companies, incorporated, and they reached out to me to work together. And it's been really productive. So I know we have a wide, diverse audience here, so I'll just define what bioprinting is for everyone. I think a lot of people are familiar with traditional 3D printing, where you have a plastic filament that's used to produce your desired shape based on a CAD file. 3D bioprinting is similar, except we're actually printing cells in a specialized matrix so that the construct isn't a hard plastic construct, it's actually living tissues. And you can incorporate other things like growth factors into these tissues as well. And you can also culture them in different types of media. And I wrote an article, a current opinion piece with Conrad and Simon, who both were founders of Aspect Biosystems, where we actually went through all the different studies for 3D bioprinting and highlighted some of their positive notes and some of the limitations in their work. So if any of you are actually thinking about doing bioprinting, here are some things you should consider. So one is your method or the type of printer you're going to use. There's a wide range of printers out there with a wide range of price costs. So you can even home make some for very cheap. You can have an Aspect printer, which is one of these high-end printers that can make clinical grade tissues. And then everything in between with things from like the BioX from CellInk, the Alevi one from Alevi, and so forth. Another important consideration is what type of cells you want to print. Most tissues, as I mentioned, the brain, have a number of different cell types. You can print primary cells versus stem cells, and also all of this is going to decide what your resulting structure looks like and also the viability. Another thing that our lab is really focused on a lot is the choice of materials for making your bioink, because a lot of the bioinks currently available were designed based to be printable as opposed to supporting optimal cell function. And so one of the things we've really worked hard on is making a really nice bioink that will print cells with high viability and function. Excuse me. And so another consideration, again, as with traditional 3D printing, what tissue structure do you want to print, and will it actually print and maintain the structure after printing? And also incorporation of growth and differentiation factors into the tissues. For those of you who want to learn more, our group has written a number of reviews on 3D bioprinting, including one in Brain Research Bulletin. And the one in Cellular Molecular Bioengineering from 2018 was actually their most downloaded paper of the year. So if you're looking to engineer any type of stem cell tissues, that's really good to look at. Here you can just see some of the different types of bioprinting. With inkjet bioprinting, you're either using thermal or acoustic forces to deposit your bioink-containing cells in your desired structure. The same applies when you use laser pulses, as you're forced to drive your structure being printed. We use a variant of extrusion printing where you can either use pressure or mechanical forces to dispense your material. And again, the type of printer and the amount of force being used is going to determine your cell survival as well as function. And then there's also cell electrospinning, which is similar to the electrospinning process. You apply a high voltage field, and then you pull out these thin strips of the bioink containing your cells. And so, as I mentioned, we were the second group in the world to get one of Aspect Systems' novel microfluidic bioprinting systems. And you can see this here, although this is getting a bit old. So they use these microfluidic print heads, and you can actually switch between 4 different materials, and they've actually increased this up to an even higher number of materials as well. And they've also recently made coaxial print heads as well. So they've got a lot of really cool different configurations for these print heads that you can use when you're making your tissues. The thing that was really appealing to us with this high-end bioprinter was that you have really good control over the pressure. So this allowed us to print stem cells with really high viability that people hadn't seen before. And we can also flip between your materials, as well as materials with different cell types. So you can actually print pretty complex stuff, and I'll probably show you some examples of that later in this talk. And when the process is really well, you can actually print drugs, which is a big benefit. So you can print 60 tissues in a day, which is important if you're going to try and screen drugs. And then this, you can just see this is their one of their tissues that they've printed, which is an artificial trachea. And if you want to read more, they actually published this last year in the FISEPI journal. And just to give you a quick history of our partnership, we initially got an NSERC Engage grant, which is one of our national funding agencies to promote academic industry collaboration back in 2016. And then the STEM commercialization to market. We leveraged this funding to get funding from Innovate BC, another NSERC grant, and a Michael Smith Pacific Parkinson's Foundation award, all to take our bioink to market. And I'll touch on that story at the end, what we've been doing with my company. One of my grad students, Laura, actually went and worked at Aspect as part of the MyTax program. And I know that also might be of interest for some of the students in India. For MyTax is a funding way to do postdocs and other research fellowships at Canadian companies. And so it's definitely worth exploring as well. We've published several papers together with Aspect, so it's been great. Conrad's a professor during his day job when he's not founding Aspect at UBC. So we've been able to publish papers together, and we actually received the top award at UVic for our research partnerships this past year, even though we weren't allowed to get the award in person because of COVID. And this is from pre-COVID times when Conrad and I got our Ignite grant, and they gave us a giant check, and it was really exciting and fun and makes me miss in-person events. So what our group has really focused a lot on, as I said, was making bioinks to enable us to print stem cells. And so we came up with a very printable formulation of the biomaterial fibrin. So when you cut yourself and a blood clot forms, fibrin is the protein that's working on it. And I've worked with this material since I was a graduate student, and 2 very talented undergrads and my very talented graduate student. So Emily and Lila Abelseth are sisters. And Laura is my former superstar PhD student. Laura and Lila actually, and I co-founded the company that was mentioned in the introduction, Axolotl Biosciences, to commercialize this product. And I am one of the co-founders as well. And so you can see here, it's a pretty complex mix of factors to get this material to both print and support cell growth. And this paper that was published in ACS Biomaterial Science and Engineering, it was actually featured in their Methods of the Year issue in 2019. So when we first made our ink, one of the things we were curious about was, were we going to actually be able to keep our stem cells alive with a high rate of viability compared to previous studies, which had only seen around 50% viability of their stem cells, which is why we printed very simple structures, as you can see here. We printed mini cylinders to try and replicate the spinal cord, and then we printed rings as well, mainly because that's something that Aspect prints a lot of, given their trachea tissues. So some work from Laura was printing these miniature spinal cords and characterizing them. And so using this system, we were printing cylindrical constructs 1 centimeter in diameter with 7 layers of fibers, an average of 176 microns using the RX1. And this came out in Applied Science in 2018. And so we're going to hit back on the things I talked about in the introduction. Laura treated them with a lot of different combinations of small molecules. So I've talked about pure morphamine and retinoic acid. And then there's also this other combination of small molecules that they use to mature these tissues to be functional neurons. And so that's what those small molecule cocktail is at the end. So essentially, she printed the tissues and then was adding these different factors to the media to see if we could change the composition of our neural tissues. So one of the things that she initially did was just take some photos. These are just phase contrast, and you can see that there are these stem cells present under the different conditions, and then they multiply over time. So this was really promising because the stem cells, these neural stem cells derived from HIPSCs, really liked our ink and were able to survive and really grow in our system. And Laura went ahead and used flow cytometry to quantify this. And so you can see we got really high levels of viability, which we really hadn't seen previously in the literature. So this was quite exciting. It really seemed like we were onto something with our bioink in terms of being able to print these stem cell derived cells. The other thing that was really interesting is Laura cultured these for 30 days, which at the time was some of the longest any bioprinted constructs had been cultured. And so they do maintain their shape, and they did it. Express mature neuronal markers. So just briefly, for those of you who aren't as familiar with neural cells, the red stain is beta tubulin 3, which is expressed by neurons. The green stain is glial fibrillary acidic protein, which is expressed by those astrocytes that I mentioned, the support cells found in the brain. Chat is a motor neuron marker, and again, Laura was trying to print these miniature spinal cords, and so that's what's shown in blue, and then they were all merged together. And you can see that the different small molecule treatments produce different ratios of these cells. And while I'm not showing the data here, you can look it up in the applied science paper, and you can actually see we quantified it via flow, showing under different conditions you got more neurons and more astrocytes depending. So this work was really exciting because it showed we could actually bioprint these neural constructs derived from human stem cells while maintaining high levels of viability, and they became mature neural tissues. And then it was interesting because we could use these small molecules to alter their composition. So we could actually tune our tissues to have different ratios of the different cell types, which is going to be important depending on the types of tissues you want to make and the types of drugs you want to screen. So, so the next thing we began looking at, or we had been looking at prior to doing our bioprinting work, is my lab had done a lot of work in the micro and nanoscale field, looking at how we could do controlled drug release to promote neuronal differentiation of our stem cells. And so those three small molecules I mentioned earlier, we had encapsulated them in drug delivery systems known as microspheres. And that's what you're looking at here is a scanning electron microscopy image of some of our drug-containing microspheres, which are small spherical particles fabricated from biocompatible polymers. And when you do them, we usually use a water-oil emulsion system. You can actually achieve drug encapsulation, and you can actually tune the drug release, which is what these three publications were showing, that we could actually encapsulate them and characterize these microspheres and then use them as a tool for making neural tissues. So here's some nice work done by my former master's student, Andrew Agby, who's actually now at Cheryl Wellington's group. So he did both unloaded and loaded microspheres to show they had similar size distributions. And then, as I mentioned, neural differentiation takes a long time. So here in his release studies, he actually characterized the release over 45 days. And this work came out in Biomaterials back in 2018. And Laura, who I mentioned earlier, who's the co-founder of my company, Laura did similar work with pure morphamine. And for both of these drugs, there was actually no published literature doing any characterization or controlled delivery of either of them. There was one paper that did encapsulation of pure morphamine, but they actually didn't quantify the release at all. They just encapsulated it and then showed that it was still effective. And again, we were making these microspheres relatively small. At the time, we were incorporating them into aggregates of stem cells, but now we also keep them small because it makes them more easy. They distribute more easily when we actually incorporate them into our bioink. And again, Laura, here in her advanced biosystems paper, we actually took this out again over 48 days. And so once we had our bioink and it was looking really good, and we found we could use these small molecules to manipulate the stem cell behavior, we actually were like, well, why don't we add these microspheres in as bioink additives to make it simpler and less steps for our graduate students? So one of the first things we did was using the RX1 was to try and show we could localize different types of microspheres to different layers of tissues. So over here, you can see a phase contrast image of 3 different fibers printed. And I mentioned that you can switch between the different materials. So in order to see a difference, because if you actually use the drug-loaded microspheres, you can't tell the difference visually between which one's containing retinoic acid and which one contains pure morphamine. So we use fluorescently labeled microspheres to see where in the tissues our different groups of microspheres are going. And so that's what Emily Abelseth did, who actually currently also works for Axolotl. So Emily printed the set of green microspheres and switched to blue and then back to green, showing we could put these different microspheres at different parts in our tissue, which eventually will let us pattern more complex structures into our 3D bioprinted tissues. And so my current PhD student, Roorkee Sharma, who's incredibly hardworking, also from India, she has been using our microsphere-laden bioink to essentially print miniature brains. And so here, one of the things that we found when we reviewed the literature is no one had actually really even tried to print anything vaguely resembling brain tissue. There was a lot of crosshatch patterns, and then Laura had printed her spinal cords. So Roorkee went for a half dome, because at least we're trying to get towards more of a cylindrical shape. And there are some other reasons for doing this in terms of diffusion and the structural integrity. But so we were the first people to print half dome shapes with this technology. And we were able to encapsulate these microspheres alongside neural progenitors. And so this came out in Frontiers in Bioengineering and Biotechnology last January. So she did a lot of work printing these miniature brains with our drug-releasing microspheres. And so in terms of the different groups, she had a group where we just applied soluble glucosterone to these tissues. She had a group with microspheres that contain no drug, and then the group containing the glucosterone-laden microspheres. And the stains you see here, TUJ1, and the first one, again, is that neuronal marker. TH is a dopaminergic neuron marker, which she was trying to do was essentially to print these mini brains with the goal we could use them to test Parkinson's treatments. And then the third channel is just DAPI, the nuclear stain. And she found that the cells really did like growing in these bioinks that contain the microspheres, and it really helped their differentiation. And so the conclusions from this was microsphere-laden bioinks promote the mature differentiation of HIPSC-derived neural progenitors more effectively than just having the soluble drug present in the media. And they also, interestingly enough, promoted increased cell survival. We're currently testing different combinations and concentrations of microspheres to determine their effect on differentiation. And Roorkee has been so hard at work, and she's got some really lovely rheological data comparing the properties of our plain fibrin ink versus the inks containing the different types of microspheres that we're writing up for publication this spring. And some of the work I'm really excited about is one of our collaborators, Hakan Nygaard, who runs the Alzheimer's clinic over at UBC. He has patients who he actually takes blood draws from and reprograms their cells back into being HIPSCs. And we've been culturing them in our lab and bioprinting them into tissues. And then the end goal is we're hoping to test some potential off-label drugs to see if they can reverse these Alzheimer's effects in our tissues. Because if they can, he can prescribe them directly back to his patients in a personalized medicine approach. And here are just some of the data showing some of these HIPSC lines that he has made from his patients and showing they can be differentiated into cortical neurons. His graduate student that worked on this project, John Frew, is defending his PhD next week, and it's just been really great to see how this project has progressed over time. Interestingly enough, these HIPSCs from his patients actually exhibit the symptoms of Alzheimer's disease in a dish. So you can see here that the amount of tau present is increasing just like it would in a patient suffering from Alzheimer's, and the same for amyloid beta. And so we can actually track these as a proxy for the Alzheimer's symptoms in our tissues. And this is just showing they make both mature neurons as well as astrocytes as well. And then this is some of the work that's come out of my lab where we actually took these cells and we 3D printed them into tissues using the Aspect system. So this work is done by John Walter Schmempke. He's an honors student who did a full-time co-op with me last summer, and he's just been amazing culturing these cells, growing them up, printing them into tissues, and then analyzing their properties, both in terms of the cellular composition and their properties with regards to Alzheimer's. So here are some of the lovely confocal images that he took. And so you can see here in his tissues, The green again is TUJ1, so we actually are getting these really long neuronal processes extended throughout our tissues. He printed at a pretty high density of 5 million cells per mil, and so these really are starting to resemble brain tissues, as you can see here. And then the blue counter stain is DAPI. And yeah, he's gotten a lot of really interesting data, and I don't have it in this talk, but he sent me this week for a grant update, and we again can also measure these amyloid beta and see increased expression of this amyloid beta in our Alzheimer's lines compared to the control lines, which again will be very important for disease modeling and drug screening applications. Okay, so with this, for the last part of my talk, I'm going to tell an interconnected story focused on something a little bit different. So Chris Lee, who graduated from UVic and worked for me for 2 years and is now my master's student who I share with Hakon, who's working on these Alzheimer's lines. When he came to my group, he was really interested in looking at glioblastoma, which at the time our group hadn't done any neural cancer work to that point. And so it was quite interesting, but he was really keen there. And so he proposed to do some work in this area. And so for those of you who don't know, glioblastoma multiforme is a primary nervous system tumor cancer that derives from astrocytes. And it's a terrible disease. If you know anyone who's had it, it's very invasive. They do chemo and treatment, and it universally recurs. So you're really looking at like maybe 1 to 5 years max of survival if you have this. And it's just a terrible disease, which, similar to some of the other neurological diseases, just really lacks good treatments. And so what Chris had initially hypothesized was that we could actually reprogram these glioblastoma cells that are highly invasive astrocytes that want to migrate in and proliferate everywhere into neurons which don't proliferate and are rather benign. And so the idea was to use this process of cellular reprogramming, where you use different techniques, and you can use this using viruses or proteins. In this case, he was focusing on small molecules. You essentially remove and remodel these epigenetic signatures, things like your DNA methylation, and this will convert your cells from one phenotype to another, with one of the most famous examples being what I talked about in my intro, where you have a fibroblast that goes back to being a pluripotent stem cell. And so in John's case, what he actually wanted to do was flip not a smooth muscle cell to a pancreatic cell, but he wanted to flip these glioblastoma cells to neurons. And so he went through the literature and did a really thorough review, and he found a couple of these small molecules. There were 5 of them, and he hypothesized that if we treated glioblastoma with these 5 molecules, we would be able to convert it into non-proliferative neuronal cells. And this work came out in 2018 in ACS Chemical Neuroscience. And so the 5 small molecules he looked at were CHIR, IX9, forskolin, IBET151, and DAPT. And so he named this cocktail based on the first letters of all these different chemical compounds. So it was the FICBD chemical cocktail. And these were done treating, so he treated human U87 glioblastoma cells. So you can see in the control cultures, the glioblastoma cells do what glioblastoma cells do, and all cancer cells do, proliferated rapidly, abundantly. Interestingly, in the small molecule cocktail, they did not proliferate, and they actually began to sort of look like neurons. And then as he got to day 7, they looked a lot like neurons, extending these really complex extended processes. And so here's just a few more of these images shown up close, where you have these processes that he was observing at day 9 and day 13. So he went ahead and did some staining to look for this. And again, you'll see this neuronal marker TUJ1. And so when you stain the glioblastoma cells, they don't express TUJ1 because they're astrocytes. Very bad astrocytes that proliferate rapidly, but they're not neuronal. But when you treat with our chemical cocktail, you actually start to see a lot of this expression of TUJ1 as well in these cells that don't typically express it, suggesting that we're actually getting cellular reprogramming here via these small molecules, which was quite interesting to see. And this was something our lab at the time was quite interested in reprogramming, and we actually had a nice paper from Megan as well, reprogramming fibroblasts and astrocytes into neurons as well. So we sort of had a little reprogramming thing going on there for a bit. Interestingly enough, so then he went ahead and did flow. And so he found that these cells that were treated with the cocktail had a much higher level of expression of this TUJ1 compared to the control cells. So this was quite exciting. It seemed that this small molecule cocktail was really reprogramming these cancer cells into neurons. To get a bit towards the mechanism, for those of you who are familiar with biology, and I know we mentioned that your university has a strong biotechnology program. So John actually looked into the levels of expression of these different transcription factors. And for those of you who are in the reprogramming field, you'll know that ASCL1, which is an analog of MASH1, is considered one of these master reprogramming factors. BRN2 and NGN2, again, for those of you who work in reprogramming, you'll know that these are, again, some of the transcription factors that are commonly used when you're trying to reprogram cells towards a neuronal phenotype. And Mary Swarning's group out of Stanford's done some really lovely work on that, where they've been reprogramming using viral vectors. But so he did see increases in some of these important transcription factors for reprogramming, alongside increases in MAP2, which is the mRNA associated with the neuronal marker expression. The other thing he did that was very interesting was he isolated the glioblastoma stem cells, which would normally form these tumors, as you can see here, these spheroids, tumor spheroids. And when he treated with this cocktail, he saw a huge decrease in the number of neurospheres formed, again, a significant decrease in the total area, and an increase in size because there were just a lot fewer of these neurospheres to measure in his work. So this was all very, very promising in terms of reprogramming and also potentially being able to inhibit tumor growth. So not content to stop there, because Chris is so good and amazing, he then decided to use our bioprinter in the Aspect system to print the glioblastoma into rings. And so here you can see some of his work from Materials Today Chemistry in 2019, where he was printing this these glioblastoma cells into rings, and he characterized their viability. The cells, just like the neural cells before them, loved our ink, loved growing in it. And in fact, being in 3D made them act just like they would do in a human brain, where they actually were very tumorigenic. And so as you can see here, here's some phase contrast and scanning electron microscopy images showing the formation of tumors in our model. And you might see some of the connections then between my work and Devka's work as well, with her cancer biology expertise and why she might be interested in collaborating with us. So again, we saw these glioblastoma cells in the presence of our ink were forming tumors just like they do in the body, which was quite interesting to observe. And also from a drug screening standpoint, it seems to be a more physiologically relevant model than, say, culturing these in 2D. And he again went ahead and characterized them in terms of their proteins being expressed. And so he found that when we printed these cells, they actually were more tumorigenic than in 2D. So they increased the expression of CD133, a glioblastoma stem cell marker, as well as doublecortin, DCX, which both have been found to be upregulated when you're looking at the protein expressions in these tumors. So this was all very interesting and promising. And then I think I leave it out because it wasn't super positive. So Chris actually used that small molecule cocktail to treat the tumor cells, and it didn't really have an effect. And I think that's partially due to the fact when they are an actual tumor spheroid, and especially in our 3D tissues, that it can't reach, it can only reach the outside population of those cells and not the inside. And so I think what happened there was they could reprogram the outside cells, but the inner cells were unaffected and they continued to be tumorigenic and continued to proliferate. The reason I bring that up is because we'll get into a potential drug that actually did show effectiveness in our model. So we published this work, and a company out of Montreal called Zonula contacted us. And so they actually do a lot of, they have a lot of candidates for potential cancer treatments. And one of the things that they had worked on was a class of NCadherin antagonists that could inhibit cancer cell adhesion, and this is all under patent with them as well. And so they reached out and they sent us this NCAD antagonist called Compound 15. And at the time in my lab, again, this was in pre-COVID times, we actually hosted a wonderful visiting student called Imke Schmitz, who came to us from the Netherlands, and she was amazing. So she came in, she saw our bioprinters, Chris was leaving, and she's like, I'm going to take over and print this cancer model. But she took it, she just really ran with it and this project, and it was amazing. So to explain why Zonula thought these incadherin antagonists would be helpful. So incadherin is a cell adhesion molecule. So it's how the cells stick together, commonly overexpressed in tumors. And so you see this a lot with the epithelial mesenchymal transition. And when you see high expression, it's going to lead to poor outcome in patient cohorts. And the more incadherin you see, it's associated with stronger glioma grades. So the idea is if you can disrupt this, you're going to cause the tumor to fall apart, and then hopefully these cells won't proliferate and they won't survive. It's sort of the idea is if you target these adhesions and then the cells don't see them and they start to die. So Imke first did what Chris did, and so she bioprinted, again, our glioblastoma cells into rings and tested increasing concentrations of this compound. And so here you can see the control cells, which again, the tumor cells, really happy in our ink, high levels of viability. And then this is after 12 days of culture. DMSO is what we use to dissolve the compound. Again, high levels of viability. And then as you slowly increased it, you can see that these cells died in the 3D constructs. So this was good. So the drug did something that was positive by killing off these cancer cells. So that was an interesting observation, and it was, validating the mechanism of the drug. And this is where it gets really cool. So she decided, what would happen if we printed a construct that was more like what you'd find in your body? And so she printed a co-culture model where she had healthy human astrocytes, similar to that you'd find in a non-cancerous brain, alongside these glioblastoma cells. And here you can see the constructs printed into rings. The reason they're red is we add phenol red to our ink during the printing process, so it helps to visualize, what the construct looks like. Also, if any of the microfluidic printheads get clogged, it's really easy to tell right away because you stop seeing the fluid flowing. Here's an image, a full-scale image of these rings. And then here's just showing the 16-day cultures. So this is also something she did that was really interesting and also really cool scientifically, is so she printed the cultures and let them all sit for 16 days. So as you can see, the tumors formed. And this was before any treatment was applied. So it's day 16, you have your healthy tissue, you have these tumors, and then she decided to hit them with a cancer drug. And I just want to point out here that she actually used a labeled version of the glioblastoma cells that were green. So it was really easy to distinguish between what our healthy tissue was and what the glioblastoma cells were, as they were glowing green. And so here you can see at day 16, everybody's got bright green tumors. Then as we apply these different concentrations, you can start to see the cells die off, as indicated by the green fluorescence protein going away. And you can see at the higher doses, we see a lot more cell death as well. And so Imke went ahead and quantified this using our imaging software. And so you can see at day 22, the two higher concentrations had really significantly reduced the amount of fluorescence. And by day 30, all the treatment groups had significantly less tumor cells compared to the controls. And if you want to read more, this is a really lovely paper that she published in biochemical and biophysical research. She also did a lot of 2D and 3D controls to show that you really needed the bioprinting and the mixture of the cells to get this really physiologically relevant tumor model printed. And also this was really great for us because it validated 3D bioprinted tissues, one, complex tissue models, and two, as a tool for screening these novel drugs. So so with that, as I mentioned, we successfully printed a novel co-culture model of glioblastoma cells in the presence of healthy astrocytes. We then used our bioprinted model to identify a novel compound that was toxic to glioblastoma, but could also shrink tumor spheroids. And we're actually currently looking to get some patient glioblastoma cell lines that we can print in 3D and then use as a tool for drug screening in our co-culture model. So with that, I just wanted to briefly talk a little bit about something that's been taking up some of my time during the pandemic that I'm really excited about, which is Axolotl Biosciences. It's the startup company that we've spun out of my lab at the research at the University of Victoria. And our goal is to sell our novel bioinks to interested groups. And so that fibrin-based bioink that I've talked about, we've been optimizing it for a number of different printing platforms, but most recently for the CellInk BioX. And so we've got our ink out with 7 or 8 different academic groups currently, and they've been able to use our base ink to print a wide variety of cells. They've been able to print human umbilical vein endothelial cells, Our collaborators in Texas have been printing functional cardiac tissue. Our collaborators at the Prostate Center have been printing functional testes, which has been really kind of crazy. And as you've seen here, we've used it to print neural tissues as well as glioblastoma models. So it's really versatile. And then we also are currently working on our second product, which is based on some of the work I showed from Roorkee with these microsphere containing bioinks that we do have a patent pending on in Canada, the US, Europe, as well as China. And so we are currently looking for beta testers. So if you're interested, just reach out to me or reach out through the company website. We're happy to talk to you and see. We have licensed the technology from the university, so we should be able to directly sell to users starting here in April or May. And so it's just been a really exciting journey with my fellow co-founders, and then Emily, who I mentioned earlier, and Kaylee, both work full-time at the company producing our ink, as well as doing quality control as well. Yeah, so here was just some of the information about us. We incorporated in March 2020, which was the week the pandemic hit and everything had to shut down. We do have a production manager, I mentioned Callie, and a quality analyst, along with a number of part-time employees who work on different aspects of the business. And we do have an informal advisory board that has extensive experience in the biotechnology industry. I did the W Venture Entrepreneurship Program in fall, which was a BC-based program for women entrepreneurs. And I just graduated this week from the Life Science BC Investor Readiness Program, which was really great. I was paired with Nancy Harrison, who's a partner at Amplitude Ventures, a VC firm here in Canada. And she's been a wonderful mentor in terms of guiding us in our company. We also, in addition to our academic beta testers, we actually do have our product out in beta with 5 different companies currently. So we're hoping to get some distribution deals in that manner. And this is Laura, my co-founder. And then this is actually Kaylee and Josh. And this was taken the week before everything shut down when we were at a tech trade show here in Victoria as well. Yeah, and just in terms of the company's vision and mission, we want to deliver the necessary products to enable bioprinting of 3D humanized tissue, which I think my talk really showed how we can do that. And really the goal is by providing bioinks that you can just essentially plug into your bioprinter and use. And the things we value are consistency, innovation, collaboration, integrity, and leadership. And it's been great being able to work with so many academic partners, and that's why we're really excited to start selling our ink, because then we can actually spread our science out to a lot more people. And if you're interested, you can follow us on all of our social medias at Axolotl and on LinkedIn, as well as on Instagram, and see some of our pretty pictures. So with that, I want to, well, you can follow me on Twitter. Also, none of this work gets done without my lab, and even in COVID times, which has been rough, they've been working so hard and so diligently to keep our science going. As I mentioned, Laura and Roorkee, Layla Abelseth, who's been amazing as well, as one of our co-founders, Imka, Megan, John, Melina, and Alex, and Chris, obviously, all been wonderful. And I'd also like to thank all of my funding sources as well. Yeah, so with that, I am happy to take any questions that anybody may have about any of this work, and I'll stop sharing. That sounds good.
Yeah, thank you, Professor Stephanie. It's a very wonderful talk, and excited to hear a lot of new things in the 3D printing area. And there are some questions from the participants they have typed. I can read it on behalf of them. So there's a question. If we encapsulate the FABCD cocktail into microsphere and print along with the glioblastoma spheroid model, will that be effective in treating the cancer cells? So this is the first question.
Yeah, I don't think so, because when we actually just put it in the media, it didn't really have an effect. It could be we need to tune the concentrations more. The thing that we actually our collaborator actually proposed is we should actually put the cocktail in with the encadherin compound, because what's happening is the encadherin compound is breaking up the cell-cell interactions. And so then we think that the cocktail could get in and actually reprogram, because the cells would be broken up. So that's one of our ideas. But along the same lines, one of our other applications is encapsulating drugs for 3D printing to test, like, because then you could test a bunch of different encapsulated drugs all at the same time in, you know, a big tissue printed construct.
Okay, thank you. The next question is, what are the epigenetic modification made by the FABCD cocktail?
Yeah, so we didn't do the necessary like proteomics to figure out exactly the regions of methylation being changed. I would guess that what's happening is we're opening up binding sites for ASCL1 to bind and then also recruit the other. So what happens is ASCL1 will recruit the other transcription factors when they actually reprogram these cells. So I think we're opening up, I think the cocktail's opening up the DNA to allow more binding. And so if you'd want to do that, you'd have to do some of your DNA mapping to see what actually is getting methylated.
Okay, so the next question is, what is the role of astrocytes in improving therapeutic effect of compound 15 in co-culture model?
Oh, they don't play, I don't think they play a role in the therapeutic. One of the important things to show that you see a lot with cancer drugs is that they actually will kill healthy cells as well. So the idea of having them both there was to see if having that cancer drug was if the mechanism was specific to the glioblastoma tumor cells or if it was going to kill all cells. And so the nice part about having the astrocytes there and surviving was showing that the drug was just selectively killing the tumors and not going to kill all of the brain tissue. So.
Okay, there's another question that how are you controlling the microspheres sizes while doing your fabrication?
Yeah, so we actually, I didn't talk about it, but there's some really exciting work going on. We currently make them in batches, and then we do SEM to make sure they're the right sizes. So we control the stirring rate and things like that to ensure they're small. But Becca Kirsch and Alejandra Fogara, who is one of Dr. Catherine Elvira's students at UVic, we actually have been working on making microfluidic chips because making our emulsions on the chips makes much more consistently sized microspheres. So we've been, so they've been working on that process. So we're hoping to switch to just using microfluidics to make our drug-releasing microspheres more consistently sized and also in a higher throughput fashion than what we currently do.
So there are some questions like when you have in the like a tumor model, like 3D printed tumor model, like can we study the metastasis, the tumors? Yeah. And could you elaborate like how we can in that the metastasis process can be tracked using the microfluidic?
Yeah, like I think using fluorescently labeled cells so you can see where the cells are migrating, especially if you have them and you can't, we didn't do this with the system currently, but you could print a tumor cell specifically into a region of healthy cells with the labeled cells in the middle, and then you could track the labeled cells as they moved. We haven't done that, but I do know other groups have. So they just use labeled tumor cells in their 3D, and then they actually track how, you know, they metastasize and where they go in the tissues. And they can print different regions of cells. So you can have your tumor cells migrating from one type of tissue to another. So I think groups out of McGill have been doing that.
And also, like you talked about the 3D printed brain. So that was really interesting. Like what are the cells we can and how it can mimic exactly like the brain? What are the things we can?
Yeah, so we have, so one of the interesting things about our 3D printed constructs, because we start from neural progenitors, is you do get, and this is in a paper that's hopefully going to be published soon. We're in our 4th round of revision. You get mature neurons, oligodendrocytes, and astrocytes. And as I mentioned with the small molecules, you can play around with the ratios. So you can actually mimic different regions of the brain depending on what compounds you use. The things we have, and again, some of this is in this paper we're about to publish, and I didn't show it today. We have been able to track electrical signaling activity, and so you can actually spike it and treat them with neurotransmitters and get changes in the electrical activity. So that's something that's important, especially if you're going to be looking at drugs that would potentially modulate that. The key features, I think, of the Alzheimer's cells, which I showed in 2D, and as I mentioned, we recently got data in 3D, being able to track amyloid beta secretion over time, and in the Alzheimer's cells, the fact that it increases over time gives us a readout that if we found a drug that inhibited that. As I said, we have this certain class of drugs from the Pressbook library, which they've been tested to be safe in humans. They don't necessarily have an application. We could actually, Hacken could actually prescribe them directly back to his patients based on if we saw the effects in their tissues that we've printed. So that's, I think, a really cool model. And, you know, there's a lot of debate in this field, especially with brain tissues, brain organoids, because some people publish them and say like, oh, this is actually generating thought. I'm not sure if we're actually there. I worry about it more than some people. You know, if we were all in person, we could talk about this over a drink because this is something fascinating. It actually comes up a lot with even like normal people who aren't in academia. The idea that you can make these miniature brains from human tissues. There have definitely been ethicists saying you shouldn't make, you know, tissues certain sizes, like past a certain size, because you might actually be getting human thought, and how would you know if that was real? On the other hand, I know some German researchers who are just like, it's all crap. You're not replicating anything. So I'm somewhere in the middle where we definitely see key features that are important for at least the outputs we're looking for. It's not perfectly replicating it, but I definitely think with aspect system and the ability to place stuff in different regions, we're going to be able to pattern some really cool things. And that's where we've gotten to now. A lot of the work here was, you know, we need to make sure the ink would keep our cells alive and they would really function. We need to be able to make sure we could actually put either the microspheres or cells where we wanted. And so now I know Roorkee's done some really interesting things where she's patterned gradients and regions into our tissues and then put the cells in. So
there's one more question from the participant, that can nanostructured material such as 2D nanosheets be used in the bio-ink as an additive to direct stem cell differentiation in absence of growth differentiation factors?
I think so. People have put in, I know there has been some work published where people have put in other nanomaterials. You often see, I think, nanocellulose gets put in a lot just to alter the properties. So it's definitely interesting. It's something we've definitely toyed a lot with. We had the microspheres already, so that made a lot of logical sense to put in there. There's also some people who will put in, you know, some of the magnetic nanomaterials as well to sort of, you know, do electric stimulation and things like that. But yeah, that's definitely a big area. And if anybody's, you know, interested, definitely reach out and we can always talk about, you know, doing projects together.
Okay, so and some more questions like what type of cross-linking is being used for the fibrin-based bioink system?
Yeah, so we cross-link with both thrombin, which is the natural enzyme, and we also have ginipin, which is a cross-linker derived from plants. And in our previous work before, we used it for bioprinting. Ginipin also is very neurogenic, so it really promoted the differentiation into neural tissue of our stem cells. So we have some of that, and then yeah, and technically the ginipin would also cross-link. We do have small amounts of chitosan and alginate just to make the fibrin itself printable, given its properties. So it could also potentially be cross-linking those materials as well.
And there's another question from the participant. Would there be any effect of loading the cells in the microsphere in order to establish the cell-to-cell connection?
We haven't, we wouldn't load cells into our PCL microspheres. I do know people do that with other biomaterials to establish microcarriers. One area where I could see it being interesting is if you were to load it into, if you were to make the spheres of the different types of cells, so then you could have, you know, patterning your regions of like oligos next to your neurons and things like that, especially as technology advances and we're able to actually place these things more precisely in our 3D structures.
And one last question from my side, like you told about your startup company, like so we are also in the process of starting our own startup company. So we need your suggestions on what are the challenges and come up with the startup company and how to make it really successful.
Yeah, so we actually have a pretty good incubator at UVic, and Canada has a lot of grant funding. So we got, as I said, all these INSERC and Stem Cell Network awards to take our product. So when we incorporated, our product was around TRL7. And so it's funny now that we have the company, and when we talk to potential investors, everyone's just confused because we actually have a product that's out being used. The biggest challenge for us was getting our license, and actually has been dealing with lawyers, which has like driven me crazy. And so, I mean, obviously COVID was a big setback, but we've done so much in a year, even with reduced capacity. Yeah, balancing your time is always a big issue. The other thing when you start up a company is like trying to figure out exactly what you want to be, and then sort of building your company in that way. Because at least here in Canada, in the life science community, it's relatively small, and so there's like certain VCs that want to see certain things, and definitely in 3D printing, it's interesting because you know, if you're familiar with the field, there's like Cellink, which is now $1 billion company in Europe, and so they actually have sort of moved away from being a bioprinting company to being like, we do everything. You'll see Aspect and Fluid Form, which are more like, we are going to print human tissues that'll go into people, which is definitely different from us, which is like, we have a product that consumers want, and we're going to sell and make money right away. So I think that that's something, depending on what your product is, like figuring out if your goal is to be like, we're a reagents company, and then I think the other things will come while we sort of figure them out. So yeah, so that's pretty much some of the things. Things I like about it is it's different. Having spent, you know, 10 years as a professor, you're used to the flow and things like that. And so I really never thought I'd start a company, but there was just so much demand for our ink because all these researchers were like, you need to sell this. Like, you can't just send it to your friends or your collaborators. So yeah, it's been an interesting ride, and you meet a lot of really cool and interesting people. So it's been nice. You know, the COVID stuff has been weird, but yeah. So yeah, that's sort of my take on it. Yeah, and enjoy the ride.
Right. Thank you very much, Professor Stephanie, for the wonderful lecture as well as the interaction with the participants through the question sessions. Thank you very much for accepting our invitation to deliver the lecture, and
Awesome.
thank you. So
Yep,
now we have our next speaker, Professor Dhirendra Katti from IIT Kanpur. Sir, Professor Dhirendra Katti, could you share the slide before I introduce you? Could you share the slide and check it?
Okay. Can you see it?
Yes, yeah. Can make it full view.
Can you see it now?
Yes, sir. Yes, sir. So, Professor Dhirendra Katti is a Girish Janakinath Chair Professor. I welcome Professor Dhirendra Katti for this one-day Indo-Canada workshop. And Professor Katti, he has received his BSc Chemistry from Pune University and BSc Technology from University of Chemical Technology, Bombay University, and PhD in Chemistry from Bombay University. Then further, he done his postdoctoral research from Department of Chemical Engineering from Drexel University, Philadelphia. And after that, he joined as a assistant professor in the Department of Orthopedic Surgery and Department of Biomedical Engineering at the University of Virginia. And later, he moved in 2004 as assistant professor in the Department of Biological Science and Bioengineering in the IIT Kanpur. And he was also head of the department from 2016 to 2019. So currently, he is a chair professor at the Biological Science and Bioengineering Department of IIT Kanpur. His research interests are in the area of biomaterials, drug delivery system, tissue engineering, and nanobiotechnology. And Professor Katti also serves on the editorial board of Journal of Biomedical Nanotechnology, Trends in Biomaterials and Artificial Organs, and also International Journal of Nanomedicine. He has received so many awards and he has a lot of publications and patents. Now I would request Professor Dhirendra Katti to deliver his lecture.
Thank you, Dr. Gopinath, for the kind introduction. So I'll get started. Is the slide visible clearly to all?
Yes, sir. Slides are visible and you are audible clearly.
Okay, great. Thank you. What I'm going to do today in the next 40-45 minutes or so is try and tell you one single story of the development of a nano-bioengineered drug delivery system. So that's the plan. And the title of my talk is a non-invasive nanoparticulate drug delivery system for the treatment of diabetic retinopathy. Now, just to get the audience to appreciate diabetic retinopathy, I'll start a little bit with the background information. So this is talking about the global statistics of diabetes. And these are the estimated number of people who are suffering from this condition. Now, if you look at the right-hand top figures, if I can, if you look at these figures on the right-hand top, you can see these are the world numbers, and the numbers are alarmingly large. In 2015, we had 415 million people suffering from this condition. And the estimate is that that number will go up to 642 million by 2040, which is 25 years from 15. And right now it's just 20 years from now. So if you look at the area where we are currently in, which is the Southeast Asian region, if you see those numbers, and if I were to make a comparison of the 2015 numbers, then you can see the world has got 415 million, whereas Southeast Asia has got 78.3 million, which is roughly 1/5 of that number. So we actually have a very significant share, which may not be very desirable, of course, but even the projected numbers for 2040 are somewhat similar in that we have roughly 1/5 of the world's population. And so going forward, it's going to be important for us to kind of put this under control. Now, what this translates into is that in 2015, we have 1 in 11 adults that have diabetes. And by 2040, it is 1 in 10. Although this 25-year transition, if you notice, it's only a single-digit change, 1 in 11 versus 1 in 10. But this advancement for 1 in 11 and 1 in 10 is very, very significant for this short duration of time for a particular condition like diabetes. So these numbers are sort of alarming, and therefore it's referred to as the diabetes emergency. If you look at worldwide deaths from different conditions, and if I were to start from the left-hand side, malaria, it in 2013 it caused 0.6 million deaths. Tuberculosis in the same year, these are all WHO statistics, caused 1.5 million deaths, and HIV/AIDS caused 1.5 million AIDS. So if you look at the deaths that are caused due to diabetes complications or diabetes-related complications, it's 5 million, which means if you put all of them together, it's even exceeding that number. So 3.6 million is all of them put together, and we're talking about 5 million due to diabetes-related complications, clearly suggesting that this is not a condition to be taken lightly. Now, going ahead, there are multiple complications that diabetes actually causes. And of them, the one of interest for this talk, my talk, is the one related to eye. So it affects multiple organs. It affects the heart, it affects the kidney, diabetes in general. And it starts gradually causing a degenerative condition in those organs. And over a period of time, if you're suffering from diabetes, let's say for 5 years, 10 years, 15 years, then these complications take over to the extent that they are the final cause for organ failure. So if you look at the eye condition that I'm talking about is referred to as diabetic retinopathy. Now, this is an image that was taken in the Kashmir Valley. And you can see on top, you have a nice view of a mountain and a valley with green trees. But the lower panel is showing you the view of a diabetic retinopathy patient. So you can see that the diabetic retinopathy patient cannot see parts of the image because of the black spots. And the other thing that you can notice is that there is a sort of a blurriness in the part which is visible. So there are two things here. One is that there is haziness in what is visible, and there are portions which are not at all visible, meaning they're black. And as the disease progresses, the extent of haziness increases as well as the number of black spots increases, and eventually leading to blindness. Now, if you take a look at this, on top you have the healthy eye, normal eye, and then on the bottom you have the retinopathic eye. And if you see the diabetic retinopathy eye, you can see that there are these large number of yellow spots, and the blood vessels are slightly increased in number. And so you can tell that, you know, the posterior portion of the eye is not at all comparable to the normal eye. Now, if you look at the statistics, eye statistics, the disease that I would like you to focus on is, of course, retinal diseases. The world population sees around 23.4 lakh people, and wherein 1 lakh is equal to 10 to the power 5. And India has got around 4.5 of those, 4.5 lakh people of those in the world, which means that we have a very significant burden of retinal diseases. And unfortunately, this condition is a degenerative condition, and it doesn't have much in terms of treatment, and therefore eventually people, you know, have to just wait until they become blind. Now, what are the current drug administration strategies? What are the treatment strategies and drug administration strategies, more specifically, available for the posterior eye? So this takes me to this very important question. Now, currently, you know, we have these 4 types of treatment or drug administration strategies. So one is referred to as the subconjunctival injection, where, you know, there is the layer called conjunctiva, and this is the needle below the conjunctiva, and therefore subconjunctival. Intravitreal is penetrating into the vitreous humor, which is this big empty hollow space in the eye. So that entry into that space is referred to as an intravitreal injection. Then there is the subretinal injection, which of course is a very sophisticated technique, and you can inject right below the retina if you're very well trained. And then there is this classical technique of systemic drug administration, which is intravenous. Now, all these 4 techniques are, as you can notice, all injectables, meaning they are invasive in nature. So the advantage of these techniques, except for the intravenous one, the other 3 which are local into the eye, is that you get sufficient bioavailability of the drug at posterior parts of the eye. However, they are also associated with significant disadvantages. The disadvantage is that injections have a short half-life and hence demand repeated administration. And this repeated administration has got side effects. So what does repeated administration cause? If you have repeated injections, it can cause pain and discomfort, of course, but more importantly, it can increase intraocular pressure and cause intraocular bleeding. And eventually it has a greater risk of infection. And finally, there is the possibility of retinal detachment. And just for the information of those who are not familiar, retinal detachment can also lead to blindness, which means that in the process of trying to provide a therapy or a treatment, you are actually causing a worse condition. Now, because of this invasive strategy of injections, these techniques have got poor acceptance by patients. And systemic drug administration, which is the last variety, it actually results in systemic toxicity because the amount of drug that actually is bioavailable in the eye by virtue of systemic administration is very less. So to make it more bioavailable, one has to inject more into the systemic administration. I mean, the systemic administration has to be on higher doses. Now, those doses, they actually lead to systemic toxicity. So therefore, drug administration in diabetic retinopathy is actually a complicated affair. On the one hand, you need to provide the drug so that you can cause relief to the patient. But on the other hand, you cannot just provide it in sufficient amounts, or you cannot give repeated injections because the patient actually has the risk of multiple other problems, that complications. But more importantly, he or she can go into the state of blindness. So the side effects of the existing drug administration strategies prompts the need for a non-invasive drug delivery system. So when we say non-invasive, what we necessarily mean is this classical mode of eye drops, what you can see on the right-hand top. of this slide. So if you can give eye drops, then that would be great. But unfortunately for us, eye drops, they're not so easy to provide for the retina, which is in the posterior part of the eye, because the eye has a large number of barriers. Now here I'm showing you 3 pathways. One is from the back, which is the systemic pathway, which is the pink arrows. Then 2 are from the front, which is the scleral pathway and the corneal pathway. The green lines are green arrows are the scleral pathway, and the red arrows are the corneal pathway. So if you provide the drug intravenously, then it will come through the systemic route, and its bioavailability is the figure at the bottom in bracket, which is 0.03%. So like I was saying, it's extremely low and therefore highly inefficient. Whereas if you go from the front through the scleral pathway, you have approximately less than 10% bioavailability. Corneal pathway, less than 1%. So based on this, clearly it feels like the systemic and the corneal pathway, because of their low bioavailability, may not be desirable pathways, whereas the scleral pathway, because of its high bioavailability, may be a desirable pathway. So therefore, we decided to look at or keep scleral pathway as one of our choices. And I'm telling you this background information because I'll gradually tell you how our choices led to our hypotheses. Now, if you look at the layers of the eye, so you can see here, this is the anterior end, and this is the posterior end. Right at the end of the eye, back side of the eye, you have the retina. Right up front is the conjunctiva. So you have conjunctiva, sclera, choroid, Bruch's membrane, retinal pigmented epithelium, and then retina. And anything that you put in the anterior eye needs to go via this route to reach the retina. By this route, I mean the scleral pathway, which we just saw on the previous slide, has the highest bioavailability. Now, on the right-hand side, you can see that some of these layers, they are permeable to hydrophilic molecules, whereas some of those layers are permeable to hydrophobic molecules. So therefore, if you were to provide either of them, if you provide a hydrophobic molecule, it will not penetrate at all. If you provide a hydrophilic molecule, then it may penetrate the first 4 layers, but then it stops after that. And these are some details on the same, but in the interest of time, I'm not getting into these details, but just should suffice for now to convey that the first few layers are hydrophilic permeable and the last 2 layers are hydrophobic permeable. Then there are a few more points to be noted before I come to my hypothesis. So mucoadhesives are known to improve ocular retention. So if you have something that can be sticky, then it would stick to the eye and therefore it would improve ocular retention time. Then there has been a study that talked about understanding various sizes of nanoparticles, starting from 20 nanometers to 200 nanometers. And they saw that 200 nanometers, they remain in the conjunctival space for almost 2 months. So which means that you can get these size nanoparticles if they are able to stick in the eye, then it would be something that can, sort of provide the drug for almost 2 months. And if you look at the last point, nanoparticles from hydrophobic degradable polymers like polycaprolactone, polylactic acid, and polylactic acid glycolic acid copolymers have sustained drug release because of their slow degradation rates. These are all known things from literature. So based on whatever I've shown you in the last 3 slides, we hypothesized that drug delivery through the scleral pathway using nanoparticles, which are less than 200 nanometers, fabricated from a hydrophobic polymer, coated with a mucoadhesive hydrophilic polymer, may enable sustained availability of drug in the retina, which is currently the major problem in the sense that everything has to be injected and you can't do repeated injections. So therefore, just to explain that hypothesis a little more clearly, this is the layers of the eye, and like I said, first few are hydrophilic permeable and last few are hydrophobic permeable. So what we have designed is a hydrophobic core and a hydrophilic mucoadhesive coating, such that this blue layer on top can allow for penetration up to Bruch's membrane. By then it degrades away. Then what you're left with is a hydrophobic molecule, and that hydrophobic entity actually passes through the remaining 2 layers and hence provides drug administration into the retina. And because it is a hydrophobic polymer that degrades slowly, it will provide sustained delivery into the retina for a longer duration of time. So that's the goal. So now what I have done is to simplify things. I'm presenting the work as a workflow and phase 1, phase 2, and phase 3 of the development of this technology. So in phase 1, we're talking about nanoparticle fabrication and its characterization, which is, I would say, the easier or the simpler part. Not too simple, but yes, relatively simpler. So I'll start with the phase 1, which is nanoparticle fabrication. So we chose, as you can see here on the right-hand top corner, a hydrophobic core, which is yellow-orange, and a hydrophilic coating, which is blue. So for the core, we chose 3 well-known degradable hydrophobic polymers, or which are polyepsilon caprolactone, poly-L-lactic acid, and the copolymer of lactic acid and glycolic acid. These are well-known hydrophobic, slow-degrading polymers. And of course, they are biocompatible, and their degradation products are also metabolizable, although they are acidic in nature. And hydrophobic cores, we use them, and we use this technique of emulsion solvent evaporation technique or nanoprecipitation for PCl, which is polycaprolactone. And we made these nanoparticles, and they were roughly 150 to 200 nanometers in diameter. And they had a negative zeta potential, all 3 of them, of around 10 to 20 in that range. Then we chose 3 mucoadhesive polymers as all of which were hydrophilic as the coating material. On the left-hand side, you can see pluronic F68, which is a mucoadhesive material, and it's also a permeability enhancer. So that's an added benefit. Then chitosan, which is a mucoadhesive polymer. And the other thing is, with cross-linking time and concentration of cross-linking, the degradation can be controlled. And gelatin, which is also a mucoadhesive material, and it has RGD motifs, which allows for cell adhesion. And also here, we can cross-linking time and concentration of the cross-linker can modify the extent of degradation, control on degradation. So therefore, we had 3 hydrophobic polymers on the previous slide and 3 hydrophilic polymers on this slide, which were used as the coating. So therefore, 3 into 3, we would get 9 systems. And so we got these 9 systems, and 3 of them are control. On top, what you're seeing are just the control. PVA is the polyvinyl alcohol, which is used in the synthesis, and therefore it's mildly coated on top. So therefore, I've just, for the sake of convenience, put it as PVA. But otherwise, chitosan, gelatin, and PF68, pluronic F68, are the 3 coating materials. And lactic acid, glycolic acid, and caprolactone are the 3 core materials. And therefore, we have these 9 particulate systems. And all these 9 particulate systems were approximately similar in terms of what we were looking for, sub 200 nanometer sized particles. When we actually measured the size, we found that you can see here on the y-axis, 250 is the highest particle size, and around 125 or so is the lowest particle size. So they were all ranging in 125 to 225, 250 range. So we said that the sizes were approximately 200, which was already reported in the literature to be acceptable. And the charges, interestingly, because of the choice of the type of coating material, we got right from minus 40 to plus 40. So we got a variable zeta potential, which was actually good for us, because if there is anything to do with electrostatics, then we would have a clearer understanding on that. And when we saw the hydrophobicity of these materials, the contact angles were roughly ranging from 30 to 80, which means they were largely hydrophilic. And the mucoadhesivity, which was ranging from 0.07 to 0.15, which is supposed to be a desirable range for mucoadhesion. So therefore, we knew that these particles, if provided as an eye drop, they would sort of go and stick to the mucus in the eye, so in the external eye, in the anterior portion. So these were the properties that we wanted to get, and we were approximately there, and therefore we were actually excited at this phase. Now, cytocompatibility is another important factor. So when we looked at cytocompatibility, we looked at a specific cell line of the eye, which is a corneal epithelial cell line, It's called SIRC. And we did the MTT assay, in which we look at cell survival, and at 24 hours and at 72 hours. And what we found was that the percentage cell viability at 24 and 72 hours was found to be greater than 80% for all the concentration, and greater than 95% for lower concentrations. So since we were not planning to use higher concentrations, it was okay in terms of cytocompatibility. So this was the first stage or phase, so to say. Then we're now going to the second stage, which is nanoparticle permeability. Now that we had made the particles based on our hypotheses, the properties were matching, and now we wanted to check if it was there was proper permeability, and if there was permeability, then what was the pathway? And then eventually, if everything is fine, then get on to the fabrication of the nanoparticles with drug inside them. Currently, they were without drug, or they were with a fluorescent molecule. So this is how we did the permeability study. So what we did was we developed the nanoparticles with a fluorescent dye, and then we put it as an eye drop in these C57 BL6J mice, and that after fixed predetermined intervals of time, we enucleated the eye, fixed it, embedded it, and sectioned it, and then looked at the sections under a fluorescent microscope. And this is what it looks like. So what you can see on the left-hand side is a collage of multiple images taken, and that's why you have these jagged edges. And this is the H&E staining of the same eye. So you can see the different layers. They have been labeled: the cornea, lens, iris, ciliary body, anterior chamber, sclera, choroid, and retina. And these are the important layers for us if it's following the scleral pathway. Now I'll give you a better view of this fluorescence, and the fluorescence, green fluorescence that you're seeing is because of the fluorescent dye which has been used in the nanoparticles. This is a magnified region of one part of the eye. So if I can go back, so if you take a look at this portion of the eye, it is a magnified region of this portion of the eye. So you can see here that the fluorescence, it looks highly fluorescent, which means that there is the nanoparticles are there in different parts of the eye. But when we did a quantification of the fluorescence, this is what we found. As we go from the front, which is the anterior, to the posterior, which is the back of the eye, because the eye drop has been given in the front. So right now on this in this cartoon, you can see that the drop would have come to the front of the eye, and then it takes the scleral pathway, which is sort of taking a curved route along this yellow colored portion, then getting into this cyan portion, and then pink portion, and then the black line. So this, if you see from the front to the back, this is how it goes, and you get a drop in fluorescence as you go from the front to the back. And this, what I'm showing here, bars, are quantifications in these boxes, 1, 2, 3, 4, separated by a mild distance. And if you look at this way, which is from the exterior of the eye to the interior of the eye, this is A, B, C quantification here, then it also shows a drop that way, which means there are two gradients. One gradient is anterior posterior, and the other one is exterior interior. So we had two clear gradients in the eye. And when we went ahead and then quantified all the layers of the eye for all the particle systems. So on the top, I'm showing the control systems, which is without any coating, and then the same PCL, PLA, PLGA particles with chitosan, gelatin, or PF68 coating. Now, if you see these, now you can see that there are bars of different colors, and they are of variable heights. The colors are each of the layers. So you can see here, cornea is red, conjunctiva is white, iris, ciliary body gray, lens is dark gray, sclera yellow, choroid blue, and then retina pink. Now, if you notice carefully that conjunctiva, the white bar, is consistently fairly tall, and so are these three bars of sclera, choroid, and retina, suggesting that these particles are more bioavailable in these layers and more importantly, suggesting that the particle is probably taking the scleral pathway. In the scleral pathway, the particle is supposed to go this route, conjunctiva sclera choroid retina. So it reaches the retina via this route. So therefore, by doing these sort of in vivo bioavailability studies, we showed that all the particles are taking the scleral pathway, that is conjunctiva, sclera, choroid, and then the retina. Now, if you remember, we had actually said that the scleral pathway has the highest bioavailability, and hence we were interested in choosing that, and whereas the corneal pathway and the systemic pathway were not desirable. So we were excited at this time during the second stage of the project that we are able to get this sort of scleral pathway that we were looking to get. So we were very excited, and therefore now we went ahead with the next step, which is putting the desirable drug into the nanoparticles. Just one point before that, we also did tissue-based separation for different coatings. So I shouldn't say separation, but quantification. So which means, again, it's an extension of the bioavailability data. So herein, I'm showing you, I would like you to draw your attention to the green bars. The green bar is actually PCL core, meaning polycaprolactone core. And the yellow bars are PLA cores and the pink bars are PLGA cores. So you can see here that relatively in the different layers of the eye, the green bars are slightly higher and many times very significant. But the most important of them all is its position in the retina. And in the retina, if you take a look, within PLA green bar, the one which was coated with PF68, it showed a very high bioavailability, almost a 20% enhancement compared to any of the other nanoparticle systems. I mean, all the systems were doing reasonably well, but on a relative scale, the PCL PF68 system was doing exceptionally great. So therefore, for our future studies, we decided to work with PF68. So PCL 68 nanoparticles showed higher bioavailability in the retina and hence were selected for future studies. Now, coming to the drug and its choice. So in diabetic retinopathy, abnormal blood vessels start to grow under macula of the retina. And these abnormal blood vessels, they are fragile and they damage the macula. So you first have, macular edema. It's known as diabetic macular edema, or DME, and which is followed by diabetic retinopathy, which is vasculature, formation of abnormal vasculature. And there is this drug, which is known as triamcinolone acetonide, which is a corticosteroid, but it has anti-inflammatory and anti-VEGF activity. And it is known to be, desirably effective for, stopping the growth of new blood vessels. Also has got a good effect on edema, but is a corticosteroid and therefore cannot be used in very high concentrations. So we decided to, choose this drug, and more importantly, the other options that are available are antibodies, or they are very expensive alternatives. And this is a chemical moiety, so therefore it is relatively inexpensive, which is a factor which can be useful in terms of therapeutics in India, or if for that matter, Southeast Asia. So I'm sorry. So we decided to go with this drug, and then we synthesized these PCL nanoparticles coated with PF68 containing the drug. And we found that after drug loading, the size was still lower than 200 nanometers, the violet bar, and the charge was still negative 15 or so millivolt, which meant that the properties were very close to what it was even when it was without the drug. We were expecting that some amount of change would occur, but fortunately for us, the change was not significant. It was very minor. So that brings us to the end of stage 2, which is now the drug delivery system is ready to be evaluated both in vitro and in vivo for the actual condition that it is meant for, which is diabetic retinopathy. So first, the in vitro data. So starting stage 3. In the in vitro data, the first and most important data is the release kinetics. And as you can see here, this is a plot of percentage drug release versus time. and the time is in days. So it released in a sustained manner for a duration of approximately 1 month. So at the end of which almost all the drug that was contained in the nanoparticles were completely released. And in the initial few days, like the first 5-6 days, we get a sort of a burst release, which is a larger amount of drug coming out in the initial few days, which is magnified and shown here. This right-hand side graph is a magnification of the first 6 days. So you can see here that, whether it is PCL, the orange line, or PF68, the violet line, the drug release kinetics is not significantly affected because of the coating. So the coating is actually not causing too much of a delay, although it is marginally behind, but it's not statistically significant. Then we went ahead after the in vitro characterization to do the in vivo studies. For this, we have used Vistar NIN, which is National Institute of Nutrition rats, which were around 3-month-old, and we developed diabetes using an intraperitoneal injection of streptozotocin, and they were allowed to the mice were then after around a week or 10 days, their blood sugar levels were checked for fasting as well as postpartum, and if they were found to be more than 120 mg per deciliter, then they were considered as diabetic. So the controls were, of course, without the injections of streptozotocin, which means that they were healthy rats. They did not have diabetes. And anytime the blood sugar level went up very high, significantly high, like 350 mg or so, then the mice were treated with insulin injections to keep the diabetes under control. Now, these diabetic animals, the diabetes in them was maintained for a period of 75 days, at the end of which they developed retinopathy in the eye. And then we treated the eye with the nanoparticles that were developed by us with other controls as well. These were the groups that we looked at. And the two main time points that we looked at is 20 days of treatment and 40 days of treatment, which you can see right on top. And there were 3 groups. We had diabetic rats where with left eye nanoparticle, right eye nanoparticle with drug, in this case TCA or triamcinolone acetonide. Second group, diabetic rats, left eye PBS, that means no treatment, right eye PBS plus drug or drug dissolved in PBS. So this is free drug, that is nanoparticle containing drug. And then the third group, which is only 3 rats because they are non-diabetic healthy rats, so we don't need too many, left eye PBS, right eye PBS. So we got double the number of eyes from this, so you need half the number of animals. So these were our groups, and PBS was at 7.4, of course. We went ahead and then did this treatment for 20 and 40 days. This, at the end of 20 and 40 days, we have done different types of characterizations, and that's the data I'm going to show now. So at the end of 20 days and 40 days, this is evaluating using a fundoscope. So the fundoscope allows you to take a view of the posterior eye, and this, what you're seeing here are those images of the posterior eye with the red lines indicating vasculature. Now, if you see here on the right, in the interest of time, now I'm going a little faster. On the right-hand side, E and J are the diabetic animals with nanoparticle containing drug, versus on the D and I, the diabetic animals with the free drug. So you can see the number of red lines that are coming out or from the center have been reduced. So fundoscopy data indicated that triamcinolone acetonide loaded PCL-PF68 nanoparticle caused a decrease in neovascularization. And then this is electroretinograms, or electroretinograms is directly talking about neuronal function, neuronal cell function, which is rods and cones. And since these cells are specifically responsible for vision in the eye, so therefore these were then looked at. In the electroretinogram, higher the amplitude of oscillatory potential, better is the function. And you can do it with dark adaptation, and then you can do it with light adaptation. When it is done with dark adaptation, it's called scotopic OPs or oscillatory potentials. So what I'm showing here is day 20 and day 40 scotopic potentials. And the black bars is what I want to draw your attention to versus the magenta or the light blue bars. So if you look at them, there doesn't seem to be any significant change. And this is in rod function. But if you look at the cone function, this is light adaptation. So rods are there in dark adaptation. Their function can be quantified in dark adaptation. And cones can be, their function can be looked at in light adaptation. So when you expose the eye to light and then do the oscillatory potential measurements, now if you see the black bars, you can see that they are significantly higher at 20 days, although they were not statistically significant, but at 40 days, it's much higher. You know, the amount, the amplitude is, the variation is almost 40% or so. So this shows that the cone function significantly improved. So electroretinogram data indicated that triamcinolone acetonide loaded PClPF68 nanoparticles caused a betterment in cone function. Now this is histopathology, which is H&E staining. So at 20 days, when we did the H&E staining of the retina, we saw that there was not much of a difference in the thickness of these layers of the or the retina. But at 40 days, what we noticed was, on the left-hand side, you're seeing the healthy animals, which is the non-diabetic. And on the right-hand side, these four are the diabetic animals. You can see that all of them have got a degeneration. And the drug-treated one is slightly better, whereas the nanoparticle-treated one was the best in terms of the retinal thickness. And this thickness, more importantly, was comparable to the thickness of the control, which suggests that the degenerative condition has been kept under control by treatment with these nanoparticles. So H&E staining data indicated that these PClPF68 nanoparticles loaded with triamcinolone acetonide showed a decrease in retinal degeneration after 40 days of eye drop administration, which was sort of comparable to the control, which is more important. Then what we did was we quantified the vasculature, which is, so what you're seeing here on the left-hand side is a 20-day characterization. And on the right-hand side, what you're seeing is a 40-day characterization. Within the 20-day characterization, the left-hand side images are low magnification images, and the right-hand side images are the high magnification images. And the white numerical at the bottom left corner of the high magnification images is the number of vasculatures that are newly formed. So you can see in control, there are only 6. Whereas PBS, there is 9, NP treated, there are 12, in drug treated, free drug treated, there is 8, whereas in nanoparticle treated with drug, it is 6. So the lower the number of new vascles, the better it is. And so now you can see that the control and the nanoparticle treated diabetic are comparable, which is where the treatment should go. So at the bottom, you have the treated diabetic mice. On top, you have the normal non-diabetic mice, and they were comparable in number of the new vasculature. So that was really very exciting for us. So retinal flat mount data indicated that these PClPF68 particles loaded with TCA showed lesser neovascularization after 20 as well as 40 days of eye drop administration as compared to these diabetic controls. So lastly, we did the molecular level characterization. So what I was showing you initially was functional characterization. Then I showed you tissue and cellular level characterization. And now I'm showing you molecular level characterization. So in the molecular level characterization, we looked at VEGF, or vascular endothelial growth factor, which is responsible for the formation of blood vessels. So here, this anti-VEGF antibody was used, and was used for quantifying the number of new blood vessels or amount of VEGF that is there. And the red color, the blue color shows nuclei of cells, and the red color is for the anti-VEGF antibody. So you can see in the diabetic animals that are untreated that the amount of red color is significant in suggesting that they would have a larger number of blood vessels, whereas the drug treated one at day 20 was not as much red, but at day 40 was quite a bit red, whereas the nanoparticle treated drug particles were almost nil at 20 days and very mild at 40 days, suggesting that they are comparable to the control. They were the best in terms of their treatment in the treatment groups. So retinal immunohistochemistry data indicated that PClPF68 particles loaded with TCA showed less VEGF expression after 20 and 40 days of eye drop administration as compared to diabetic control, which is NP alone or drug alone. Now, in the molecular level data, we have another point that we wanted to look at. This is referred to as GFAP, or glial fibrillar acidic protein. Now, this protein in non-diabetic conditions, it is mostly limited to the proximal retina, whereas in diabetic conditions it spreads through this entire region of the retina. So if I show you the characterization, it will be much clearer. So here I'm showing you GFAP antibody, which is green in color, and here you can see DAPI is for nuclei, GFAP is here, green color. So you can see it's only in the proximal retina and does not stain in other parts, whether it's day 40 or day 20. Whereas the diabetic animals, you can see that the GFAP is not limited to the proximal retina, but spreads through the entire retina. Same is the case with NP, which is blank nanoparticles. In drug treated, there is some mild effect at the 20 days, but it is not so pronounced at 40 days. In nanoparticles with drug, this effect is sort of comparable to free drug at 20-day time points, but at 40-day time points, it is more proximal and does not show a spreading through the rest of the eye, because you can see the DAPI staining right here at the bottom. So again, GFAP limiting itself to the proximal region also was a very good indicator and showed that these nanoparticles loaded with triamcinolone acetonide showed less GFAP expression after 20 and 40 days through the entire eye and was limited to the proximal region. So taken together, this study, I can now come to the conclusions that we developed these core shell nanoparticles of approximately 200 nanometer size with different physicochemical properties were successfully fabricated, and all these fabricated nanoparticles, they took this scleral pathway, which is conjunctiva, sclera, choroid, and retina, and the PCL core showed more bioavailability in all the layers, and hence it was the one which was selected with preferentially the PF68 coating. Now, different shells have different distribution in specific layers of the eye, which is to indicate that nanoparticles have got variable tropism in eye tissue. And pluronic F68 coated PCL nanoparticles preferentially accumulated in the choroid and retina. It was the one which showed the highest availability in the retina, and hence it was the one which was evaluated. And at the end, TCA, which is triamcinolone acetonide loaded polycaprolactone PF68 nanoparticles, showed promising results for the treatment of diabetic retinopathy. Before I close, I want to acknowledge my funding agency. This project was funded by ICMR as well as DST Nano Mission now. And Beena Pani on the right-hand side was the student who did the work, and Srinivas Rao, who is continued the project now. And Dr. G. Bhanuprakash Reddy at National Institute of Nutrition was of great help for conduct of some of the studies which were done at his lab. And for this work, Beena Pani received the ISCA Young Scientist Award from the then Vice President, Mr. Hamid Ansari. And this was published in nanoscale as a cover. So with that, I want to thank you all for your attention. Happy to take any questions. Thank you.
Thank you very much, Professor Kati. It was a really wonderful talk, and it was exciting to hear about the diabetic retinopathy and animal model studies. There are a few questions in the chat box, which I'll read it on behalf of the participants. So there's one question like, why only 20 and 40 days were only chose specifically? Why not early or latest days?
Okay, good question. So if you remember that we had done an in vitro release kinetics study, and in the release kinetics, it was seen that the drug was being released from these nanoparticles by approximately 30 days. So the reason we chose a few days ahead of 30 and a few days after 30 was to understand whether the drug has its effect even early on, or it needs to have complete release before it can have its effect. Hence the 2 time points, 20 and 40.
Okay, there's another question like, how did you check the mucoadhesiveness of the nanoparticle?
So this is a standard assay in which you can conduct the mucoadhesivity test, it's called. And from there, if you are developing anything that needs to stick on mucus, then you need to do that assay. So you can just look it up in literature, you'll find it. It's not a big deal.
Okay. And there's another question from the participant. What pathway does the drug follow to reach the retina?
So the pathway is referred to as the scleral pathway, and the actual tissues that it experiences before it reaches the posterior eye is choroid, sclera, and then retina.
Okay. So there's another question that what was the reason that the nanoparticles specifically took the scleral pathway to reach retina?
So the hypothesis, yes. So the hypothesis was that if you remember, I showed the entire layers of the eye, and the initial few layers of the eye are permeable to only hydrophilic molecules, and the latter layers of the eye are permeable to hydrophobic molecules. So using that to our advantage, we decided to take particles such that there is a core and a shell, and the shell is hydrophilic, it degrades away, and then remains the core, which is hydrophobic, and then it will go in. So we have made a design, so very pertaining to this workshop, nano-bioengineering. We made the design such that it would take the pathway that gives us the highest bioavailability. Hence, it was designed that way, and it took that path also.
Okay, in continuation to the same question, that is there any surface chemistry plays the role in this for favoring that route? Or
Sure, yes. I mean, the mucoadhesive layer that is there is playing the role of, I mean, the surface chemistry there is playing a role. It's a very fine coating, so it's not like a thick layer or something like that. So it is surface chemistry that matters.
And you also told about that one of the microscopic technique to understand the retinopathy. Could you add a brief on that?
Which microscopy? Are you talking about fluorescence microscopy?
Oh no, there is a
Fundoscopy?
Yeah, fundoscopy.
Fundoscopy is in a live animal. If you want to see the posterior of the eye, then in that case, this is the standard technique that is used even in human beings. So the very same technique is used for visualization of the posterior eye in color.
Okay, okay. Is there any other question from the participants? I think. Okay, sir. So I think almost all the participants have typed their questions. So if there is no questions, then let us thank Professor Kati for his wonderful lecture. And thank you, sir, for your wonderful talk. We'll meet you soon.
Thank you for the kind invitation.
Sure, sure. So
okay, so can I stop sharing now?
Yes, sir. Yes, sir.
Okay. Is it okay if I leave?
Yes, sir. Yes, sir. Thank you very much, sir. Thank you very much.
Thank you. Thank you.
Professor Devika. Yeah, could you share the slides before I introduce the?
One second. Can you see?
Yes, yes. Very good morning, Professor Devika. Thank you for accepting an invitation. And the next speaker is Professor Devika Chitrani. She was awarded the faculty gold medal and the gold medal for physics when she received her bachelor's degree. She was awarded with many fellowships by Natural Science and Engineering Research Council of Canada to conduct her graduate and postdoctoral work at University of Victoria and University of Toronto, Canada. So following a successful completion of her doctoral work, she was awarded one of the most prestigious awards in Canada, that is NSERC-PDF, to continue her postgraduate research in the Department of Biomedical Engineering at University of Toronto. Now she is an associate professor at University of Victoria and directs the state-of-art Nanoscience and Technology Department. She is also part of the medical physics faculty at the British Columbia Cancer Agency. Dr. Chitrani leverages nanotechnology to create innovations that advance the care of cancer patients. She is using gold nanoparticle as a radiation dose enhancer in cancer therapy. This work is featured on the cover of Radiation Research Journal and was also awarded the Michael S. Patterson Publication Prize in 2018 by the Canadian Organization of Medical Physics. She has developed three-dimensional tumor models to optimize bio-nano interface in cancer therapy. This work is featured on the cover of Nano-Microletters Journal. Dr. Chitrani is considered as one of the leaders in the field of nanotechnology, and her publication have received more than 10,000 citations. Dr. Chitrani has earned a strong international reputation for her innovative research through her many review articles, book chapters, and invited presentation. Her passion is to develop smart nanomaterials to improve exciting cancer therapy. Thank you very much, Professor Devika. Please, the stage is yours.
Thank you so much for the invitation. And so today, this is more educational. My talk will be for especially for graduate and undergraduate students. So what I'm going to talk to you today is how we can cross barriers. If you were to use this nanotechnology in basically cancer therapeutics, that will be my focus. So when I want to talk about nanotechnology, so I would like to give credit to Richard Feynman. He is the actually first one to unveil the endless possibilities of nanotechnology. And he's a Nobel laureate too. So I always show this slide, to show the appreciation for what he has done. So now I don't think, we need this slide, but just to be sure and everybody is on board, I just want to explain what the nano scale means. So for example, if you take a drop of blood, so it basically has 5 million red blood cells. Think about it. And then this is a red blood cell. The diameter of a red blood cell is 2500 nanometer. And if you look at the width of the two complementary DNA strand breaks, it is 2 nanometer. So now you should have a feeling of what's the scale of nano. Now, today I divide my talk into a few sections. First, if you were to overcome barriers, so how do we overcome them? And what are the barriers? So I started with the individual cell. So this is a nucleus of a cell. And think about it is like an egg, right? And this is the eggshell, that is the cell membrane. And this is the egg yolk. Basically, that's the nucleus of the cell. And this is basically the cytoplasm of the cells. So if we were to use these nanoparticles for therapeutics, so you have to actually see how these nanoparticles get into the cells. How can we optimize this process? That is the first step. I will talk to you about that. And the second thing is it's good you look at single cells, but you have to think about when you talk about a tumor, it's a tissue, right? It's a 3D structure. So it consists of so many cells. So now the question is, whatever you find in your individual cell, how can you relate into a tissue? So how do you develop these models in your lab? So I'm going to talk to you about that. So to troubleshoot your experimental therapeutics. And finally, when we talk about a tumor, is it just a tumor cells? So it is not. So we have to think about tumor microenvironment, which consists of other cells as well. So if you are to be successful, you have to consider all of these options to come to a good therapeutic approach. Now, in order to do that, I actually select gold nanoparticle as a model system. So when you think about gold, you think it's a yellow, this is a gold, it's yellow, but when you make gold nanoparticle, actually it turns into this red wine color. So what you do in this process, you change the properties of bulk gold to gold nanoparticles. So it has certain features that we can use for applications now. So now, the gold nanoparticles provide lots of opportunities. As I told you, I'm a professor at University of Victoria, so I do fundamental research. I also do applied research. Fundamental research is very, very important. Applied research is also important because that's when a master's student can go to a PhD, right? So to applied, and then you can get funding in 2 agencies, like for example, in Canada, NSERC is more towards fundamental research, CIHR and other granting agents are more towards the applied research. So you need to have both, and that actually benefits your graduate students in your lab and at your university. So in terms of goal, the reason is it's very easy to make them. I tell people it's like you making tea. So you change the ratio of sugar to milk, you get a different flavor. Similarly, when you make gold, you make the ratio of specific reducing agent to the gold salt, you end up with different sizes of gold nanoparticles. Very easy to do using the same system. So you can do fundamental research with it. I will show you in a minute. And of course, I'm not a chemist, but the surface is easy to manipulate. So you can put ligands very easily. Surface chemistry is not that complicated. And the other thing is the biocompatibility. If you were to use it for biomedical application, is it biocompatible? Yes, the phase 1 clinical trial has finished with gold nanoparticle and shows the biocompatibility of it. So that's very good. And the other thing is it has applications in, I will talk to you about, I am more focused on using gold nanoparticle as a radiosensitizing agent and also as an anti-cancer drug carrier. So that actually combine 2 approaches in cancer therapy. That's a very important thing. So now I'm going to answer the first question now. How can we optimize the gold nanoparticles uptake in these cells? Or the other way I ask you the question in a different way. Will the size of the nanoparticles really matters? What do you mean by nanoparticles here? What is the size? So we talk about 1 to 100 nanometer, right? So it's a very small range. You may think that I don't think it will make much difference. It does. So you will see what happens. So just to give you a small tutorial, these nanoparticle actually mostly enters the cells, something called receptor-mediated endocytosis. So it actually bind to these receptors and it get actually invaginate like this and then end up with these endosomes. These red particles are nanoparticles here, and then fuse with lysosome, it get excreted. So the question is, will the size of these nanoparticle really matters how they accumulate in these cells? That was one of my earlier studies where I actually use, you can see different size gold nanoparticle, 10 to 100 nanometer. You can make it easily. And then I found something really interesting. What was that? That is, there is an optimum size, which is 50 nanometer in diameter, has the optimum uptake in cells compared to smaller and larger. So you can see from these images, you can see the nanoparticle is at the cell membrane here. So you can see it here in this TM image. So here it shows that invagination. So you can see that the dark dot is a gold nanoparticle, and you can see they are trapped in these organelles here. Same thing here. It's a trap in these organelles. The black dots are gold, and at the end it gets excreted. So this is what I see here. So this is actually this paper. This is my first paper in biology because I did my PhD in quantum physics. First paper in biology, and then this has over 4,500 citations. Can you believe that? Yeah. So this is a good message to PhD students or any other graduate student is when you do your PhD, what you do here is you learn how to think. So this is what I did my postdoctoral work. Biology, I haven't taken a single biology course, but I learned everything, and then at the same time you produce something really good and really valuable to the community, right? So that's very important. And so as a physicist, I always think like that. What is the reason behind what I saw with the size depends? So I came up with the model to explain it. when the nanoparticles are really small, the membrane has to wrap them. It's like a gift wrapping. It's very, very difficult to do. So they have to cluster together. So it is a probabilistic process. But when the nanoparticle is big, what is happening? It can easily wrap by the membrane. But the problem here is that it actually takes a lot of receptors on the membrane. So there are a lot of areas without no receptors. So even if a nanoparticle comes, it cannot make the contact and go in. So in the middle, there is a size that actually is favorable. That's why we see a smaller, larger here, and this is 50. So there is a favorable size in the middle. That is my explanation for that. And I wrote a review article gaining much attention as well. Summarizing all of these findings, I did a lot of things, but I'm just giving you a flavor of what I have done. So now, talking after you talk about, okay, so the gold nanoparticle has a this uptake dependence as well. But then as soon as I finished that one, I joined Princess Margaret Cancer Institute to do radiation research. The reason was gold is basically a radiosensitizer. Why? Because in the presence of the gold, what happened is the radiation can produce more free radicals, enhancement in the production of free radicals. So these free radicals actually can damage DNA. That's how it actually acts as a radiosensitizer. I'm going to give you a small explanation about what is happening in radiation therapy. So the goal of radiation therapy is basically to kill the tumor, right? So the radiation is focused. So what we do, say, think about it. Here is where the the tumor is. So you can see when you use a linear accelerator, it rotates. The idea here is that you send these photons, which are X-rays, from different directions. Basically, you are targeting the tumor, but try to go through the normal tissue not more than one time if you can, because you don't want to damage the normal tissue. So there is actually, you have to be very careful the normal tissue toxicity versus controlling the tumor, probably controlling the tumor damage as well. So you have to interplay these two. So what is happening here is if you look at this graph, this dotted line is the normal tissue complication probability, damage to normal tissue. And this is the tumor control probability. Now think about these two now. So think now you want to achieve 80% tumor control. So 80% tumor control means here, right? So that means what is happening here is so you have to apply this dose to do that. And then but it actually produced, this is the normal tissue complication, right? So it produced 50% normal tissue damage, which is not good. So what we want to do is to put a radiosensitizer in the tumor. What it does is that it enhances the local radiation dose. So what it does actually, it pushes this tumor control probability curve to the left like this. Then see what happens. So now, if you want to get the 80% tumor control, 80% tumor control. So we are talking about this one now. So you can see, look, 80% tumor control, the normal tissue toxicity is about 5%. So that's the goal of having a radiosensitizer. That's what it does. That's what I want to get to you, what it can do. So now that's the theory behind. So now, when I joined the Princess Margaret Cancer Institute, so what happening? There was some research done in the field. So Hainfield actually showed if you use gold nanoparticle with radiation, you can actually kill basically the tumor cell. Then the tumor volume almost reaches 0. But there were some drawbacks of this study. The one is the concentration was very high because he used 1.9 nanometer gold, very small size gold, and he used low energy radiation. Low energy radiation, you can't use it to treat patients because low energy ones cannot penetrate if the tumor is deep seated. So you have to encounter two things. One is you have to do your research at linear accelerator, that is MEV energies, and also at the same time you have to reduce the amount of gold that they will have to use. So by looking at what I have done before, I realized the outcome of this very easily, because if you look at it, he used 1.9 nanometer gold. So here it is 14. So 1.9 nanometer gold is somewhere here. Uptake is very, very low. So that's why he had to inject very high amount of gold. So what I have seen in my studies is I was able to reduce the concentration of gold by a factor of 10 to the power 6 now from this point, by just optimizing that type of, uptake at cell level. And we will talk about these things. And just to give you an idea why it's important to do this kind of modification to cross some of the barriers that what we see right now. So now what I did was I actually used 3 different sizes of gold. For example, I wanted to prove my concept, right? So this is actually ortho voltage unit. This is used to treat skin cancer. This is low energy. This is where the petri dish cells are there. Nanoparticles are in those cells. And then you give different amounts of radiation dose. So this is called the survival of cells. And then you can see by look at here, it's easier to follow. So this is actually cells with gold nanoparticles of size 50 nanometer. It has the highest uptake, so it has the lowest survival. This is the control with no gold. This is smaller and larger. So this is what you expect, right? From our basic study, we know 50 nanometer particles goes into the cells very effectively. So that means it will produce more sensitization. It leads to lower survival. So that actually now you can connect the dots, right? And then this is actually looking into the survival of cells. I also look at, because our target is the nucleus, right? So what I did was I look at the DNA damage. So in this case, these round ones are basically the nuclei of the cells. And so what happened when there is a DNA damage, the DNA get prepared, right? So then there are some proteins that come for the repair process. So you can actually tag these things with fluorescent proteins, and you can see I track 2, and you can see the top one is with no gold in cells. This is with gold in the cells. You can see these all foci represent damaged DNA, double strand breaks, because that's the most lethal one. And so there are some theoretical, there are some other studies to prove what I have done. That is, but this is done with the DNA films with gold nanoparticles. What it shows is that when the DNA is incorporated with gold, you get a higher amount of DNA double strand breaks with the DNA with no gold actually. And the mechanism I told you before is the gold enhances the production of the free radicals that can damage DNA. Now the question is, will it hold at this energy? That is, this is a linear accelerator. If you haven't seen it, this is where the patient lies. So I put my samples there, and this is solid water. You do a treatment planning, you do your experiments, and what I noticed was that there is a DNA damage, increase in DNA damage with gold nanoparticles. For those 2, you can see track with 2 repair proteins. You can see enhancement in the DNA damage. So what you see here is the top one with no gold, this one with gold. You can see the enhancement in DNA damage foci with nanoparticle versus with no nanoparticle. This is the merged image of the 2 repair proteins, right? And this is the one actually featured on the cover of the radiation research, and it wants the Michael Patterson Award as well because of the impact it made in the field of radiation research, right? So this is basically a 3D reconstruction of a nucleus. All you can see is the foci are the damaged DNA actually. So now, if you remember, we talk about that. If the gold nanoparticle can create these free radicals and does the DNA damage. So the question is, previously the nanoparticles were not in the nucleus, right? So the question is, if you can target them into the nucleus, you should see more damage. That's an obvious question, but it's not easy to target them into the nucleus because nucleus has pores which is almost closed. So like it's a 2 to 3 nanometer, but if you were to send nanoparticles, you have to open them up. So they can open up to 25 nanometer through active targeting. So this is, I published in nanoscale as well. So you can see, and then what it is not easy to target them into the nucleus, because you have to get these for example, nanoparticles enters through endocytosis process, trap in these organelles, but then you have to get them out of these organelles to the cytoplasm if you were to target them into the nucleus. So this is, I'm showing, they come out of these organelles, and then here, this is the nuclear membrane, this is the nucleus, mark it in here. They are waiting to go in, and then here it shows this is the nucleus, they go inside the nucleus, and then so we were successful in targeting them into the nucleus. And we of course look at the radiation. What we see was there is an enhancement in the cell survival fraction was reduced quite a bit with nuclear targeting versus non-nuclear targeted ones. So that is very good. And you can see the DNA damage increase here compared to non-targeted one. But now the question here is this: targeting the nanoparticles to the nucleus is very specific, right? So you need to find the vectors that targets them into the nucleus. But I was looking for another option, which is basically I can use it for any patient. So it is not patient specific, if I were to put it that way. So what I did was I used another drug called docetaxel. I don't know, you heard about that. So docetaxel is a drug actually, which is also a radiosensitizer. You have to think about that. This is a radiosensitizer as well. The gold nanoparticles is also a radiosensitizer. So what docetaxel does is remember you have in the cell, you have this marked in green is the microtubule network. So that is the highway of the cell. So the marked in red here is the nanoparticle clusters going along this microtubule network. So what happened when you put docetaxel into the cells? So the microtubule network get stabilized, right? So so then what happened is that as a result, most of the nanoparticle get pushed towards the nucleus. This is exactly what I wanted. They don't have to be in the nucleus, exactly. So then when you expose them to radiation, it creates a shower of these secondary electrons that actually interact with water and produce these free radicals, right? This is what you want. So you expect a increase in damage of the DNA and the lower survival when you treated cells with docetaxel. Is that what you see? We will figure that out. So now, there's another important thing that docetaxel does is it puts these cells in, this is the control cell, the dotted one. When you put docetaxel, it blocks the cell cycle in something called G2M phase, which is most sensitive to radiation. Now think about it. The cells are, this docetaxel puts cells in a phase which is most sensitive to radiation. And so then you have a physical radiosensitizer like gold. So then you can see you expect a synergistic effects. That's in that way because their mechanisms are different. Is that what you see? Yes, that's what we see. So we also saw something very interesting. This is synergistic effect. This is just the docetaxel with radiation. This is docetaxel gold nanoparticle with radiation. You see a very good effect. The other thing we noticed was the retention of nanoparticles was much higher with docetaxel because docetaxel is given to patients weekly, right? So if you inject gold once, then we expect gold to stay there because as long as the effect of docetaxel is there, nanoparticle can stay in the cells because the microtubules are not free to put them outside the cells. So that's very important. And this was, we published it in British Journal of Radiology. And this is very interesting moving forward for us. So now the question is, we use the free docetaxel with free gold, right? So now there is a recent paper, it's from Shankaran group, just came out actually. They are showing how this docetaxel can be placed on the gold and then do this. So that way we can actually go one step further where we can actually use gold nanoparticles to deliver docetaxel, and the gold can also act as a radiosensitizer, and that means you can have a controlled delivery. If you think about chemotherapy right now, what's happening is only probably 0.1% of the chemo drug gets into the tumor. So you may ask a question, do you think gold nanoparticles can get into the tumor? I will explain it to you that soon. They can. So now I just want to talk to you about, so we talk about radiation therapy. Gold is a radiosensitizer, and gold actually can be used to deliver drugs too. So that means I should be able to make combine the two with gold to make better to have actually better outcome in radiation therapy. That's the idea. Of course, people are trying other options too. I wrote a review article about that as well. So you can take a look at it when you have time. So far I focus more on 2D monolayer of cells. So now our second goal is what happens to these nanoparticles in tissues. Very important, right? When these nanoparticles go through the blood vessels, the leakiness of the tumors allows them to get into tumor tissue, right? So the question is, can they penetrate through the tumor tissue and reach individual cells? So how do you know that? Before you do animal studies, it's very important, right? So what we do, we grow these 3D tissue structures in our lab. This is a structure, so you can see C cell, so the top to bottom. So we grow these tissues and we look at the nanoparticle transport through these structures. So this is a monolayer of cells. There is no extracellular matrix very much, but it is a 3D tissue. You can see extracellular matrix. This is all gold marked in yellow here. And you notice something very important here. What do you notice? This is a monolayer cultures. Smaller, we use two sizes, 20 and 50 nanometer. 50 nanometer has the optimum uptake. We know that, right? But in the tumor tissue, what we noticed was the small nanoparticles are better than 50 nanometer to a certain extent. So what we did was then we had to go back to the drawing table again, because if the small nanoparticle penetrate through the tissue better, then what you want to do is that when they reach the individual cells, you want to increase their uptake like 50 nanometer, basically. You want to increase the uptake, otherwise what's the point, right? And then what we did was that we actually added a driving force like RGD, which is an integrin binding domain, which is available on most of the tumor cells to enhance the uptake of small nanoparticles into the cells. When they reach individual cells, they penetrate through very well. So what we noticed was we noticed something interesting. So we were able to increase the uptake of small nanoparticles at monolayer level and also at the tissue level, because it penetrates through tissue very well. So that was important. And then what we did was okay, now we tested our nanoparticle system in the monolayer level and at tissue level. So the question is, will it work in an animal model? That's what we tested. It's very important, right? So the way I do it, I test it in these models before I test it in an animal model. So what I noticed was something very interesting. That is, 10 to 12% of the injected dose gets into the tumor, which is very high actually. If you look at published data, it is very high. So that is because of the optimization I have done using these other interfaces. It is very critical. So if you look at it, this is tumor tissue, right? So this is actually a blood vessel cut open, you can see. And these are openings of the blood vessels. And these are tissues. You can see gold in the tissue as well. And then within 24 hours, you reach 10 to 12%, which is ideal for our work. And then this is actually silver stain, just to show the gold is dark actually in a tumor tissue, which is very good. Now, that is also good. So what I wanted to touch base with you is some other aspect. Like when you talk about tumor, are you thinking about just tumor cells? That is a big question, right? Are we just thinking about tumor cells? No, because you have to think about tumor microenvironment, right? If you were to do better therapeutics, so what we found was something very interesting. For example, if you look at a tumor microenvironment, in addition to tumor cells, it has two other important components. There are other cells too, but our attention went to fibroblasts. Any tissue has fibroblasts, right? So what the tumor cell does during the initial processes, it actually convert these fibroblasts, recruit them, and then convert into something called cancer-associated fibroblast. So what the cancer-associated fibroblast does, actually it supports tumor growth. So if you want to make better therapeutics, you not only have to target tumor cells, you have to target CAFs as well. Of course, fibroblasts suppress the tumor growth. So the question is, what is the ideal situation here for me? So I want to target gold into tumor cells and also to this CAF. But I want the uptake must be less in fibroblast cells because I don't want to kill them, right? So the question is, so what we did was we look at to make it simpler, individual cell cultures, tumor, fibroblast, CAF, and we did the uptake, we did radiation, and we look at the damage. Let's see what we got. We got something really interesting, very encouraging. That is, the CAFs and the tumor cells take more nanoparticles compared to normal cells, which is very encouraging. You can see that these are tumor cells marked in reddish gold nanoparticles, and this is CAFs. You can see very high uptake in those cells, and these are normal cells. Uptake is much less. So as a result, you will see with radiation, you saw more damage in CAFs and tumor versus normal cells, which is very encouraging for us. And we publish in Nature Scientific Reports. You can read about it. And then so we touch three bases, right? How do you optimize at single cell level, tissue level, and also now we talk about animal studies, and we talk about tumor microenvironment. Now my goal is basically to use these gold nanoparticles actually in combined therapeutics, and which is radiation therapy, chemotherapy combined in the presence of the gold. So in order to do that better, I have proposed a couple of platforms. One is the lipid nanoparticle platform. That is, we put gold and the drug in this lipid system. So we have been successful just putting gold first, and but we are incorporating the drug into it. But the only problem right now is to trigger release. That's we are working on right now. That is one system. What is the other system? Oh, sorry. So I forgot to tell you that the nanoparticles uptake, you should be able to increase by putting them in a lipid system, because small nanoparticle uptake can be enhanced. It's like I call it Trojan horse delivery. You can do that. And the last one I want to mention is this is another one. We have a pattern on this one. Is you use actually, the Hainfield has this nanoprobe, and they make this gold lipid. You can exchange the bilayer, sorry, the lipids in the bilayer with gold lipid, and our goal is to put the anti-cancer drug in the core. Actually, that's another option. You can see gold nanoparticle uptake can be increased by using this kind of a system, and it can be a controlled delivery. And so the last question here is, can I do it alone? No. So I work with a lot of researchers, and for example, Sunil Krishnan is a clinician who is working with gold and gold nanoparticles, and he's a clinician at the Mayo Clinic. I work with him, and he's a good collaborator. And this is Jan Schumann from Harvard Medical School, and he does a lot of Monte Carlo for my work. You can see my publication. He's from Massachusetts General Hospital, and he's from MD Anderson Cancer Center, and he's from Queen's University in UK. And so likewise, we have a lot of, so we talk about how we can translate this work into the clinic. And we wrote another article recently, which has a lot of people in the field who are expert in the field got together, and then we talk about how do we do that, and we actually try to put together all our efforts, so that we can do this in this lifetime. I mean, that's the idea. So there is nothing to be scared. You share your ideas, and we are actually working on putting grant proposals together, and these are my heroes. So a lot of people, a lot of young scholars and granting agencies. I'm very proud of them, and they do all the work, and while I get to enjoy everything else. And so this is my mentors, Dr. Richard P. Hill and Dr. Leon Sanch. And I think I'm going to stop here. Thank you very much for listening to me.
Thank you, Professor Devika, for such a wonderful lecture. And now I would share some few questions from the participant typed in the chat box. So the first question is like how the gold nanoparticle directed towards the tumor regions by the process of radiation. I think he's
Right.
trying to
So,
ask like
right.
how it is
So,
targeted.
Sorry?
Yeah. I think he's trying to ask how it is targeted.
I see. That's a very good question, actually. So what I did was these gold nanoparticles are actually functionalized with something called RGD peptides. So this is actually can be targeted to blood vessels and it, as well as to the tumor cells. It's more or less overexpressed in tumor cells. So idea here is the gold, and once you inject these gold nanoparticle intravenously, so what it does actually, it travels through, right? But if you have to actually cover them with the PEG, if you know polyethylene glycol, that's why we have to put this RGD peptide. It's a natural peptide to increase the uptake because a PEG reduces the uptake. But the idea here is that then the tumor blood vessels are leaky, right? Because it forms really fast, so it's very leaky compared to other regions of your body. So that's how, if we can have the circulation long enough, you can actually get these nanoparticles into the tumor. And then the question is, can they go through the tumor? That's why I said we did a lot of tissue work before I start the animal work, right? My first animal study worked very well. That's what we published. And now to answer the other question is that we are actually moving into patient-derived tumor models too. That means you get tissues from your patients and we grow these things in patients, in mouse models, and to do studies where you really can see real tumor microenvironment, which you may not see if you use standard cell lines. So to explore these options of CARs, fibroblasts, and so forth, and how it's going to interact with your therapeutic options and things like that. Good question, yeah.
And the next question is like, will the radiation not affecting the neighboring cells?
Very good. So, very good question. Yes. So the idea, I think I told you in that diagram. So the idea here is that as you saw that normal cells are not taking up gold very much, right? Which is a good thing. So what we are doing here is that we are trying to increase the dose within the tissue, tumor. So we are targeting the radiation to the tumor too. So when you have gold nanoparticle in the tumor, I think I showed you in that diagram, for example, I think, can you see it now here? So yes. What we do is that this tumor control probability curve is actually shifted to the right, if you can see. So to get the 80% tumor control, you can see the dose is much less compared to before. This is the before when we didn't have radiosensitizers. So that means it is a higher dose. So you reduce the dose, you can see normal tissue toxicity damage is much less. And when we have less number of gold in the normal tissue, it's even better, right? That's what we saw. That's why we think this will be very promising. And actually, we already started, we got the funding and we are doing animal studies. As we speak, we are designing all of these studies.
Okay. And the next question is, after entering the cells, how do the gold nanoparticle escape the endosomes to get released into cytoplasm for further action?
Very good questions. All of them are good. So that's a very good point. Is if you remember when I was talking about nuclear targeting, this one, right? It's this one. So in the case of nuclear targeting, remember I told you so that we have to functionalize gold in a way. So these once they get in, they're trapped in these endosomal vesicles, right? So if you were to target them into the nucleus, you have to get them out of these endosomes. So how do you do that? That means this RGD peptide has this capability of actually escaping these endosomal vesicles too. So I'm not targeting them into the nucleus, but by doing that, that is why actually you see that retention of gold nanoparticles is much higher even without nuclear targeting. In most of my recent work, I have talked about this one, why it's the case, and it actually gets into the cytoplasm. So, and then that actually allows them to stay longer and accumulate in the cells without getting out it very quickly so that you have enough gold in the cells to cause enough damage to, during the radiation.
So the another question is like how this protein corona affect this, because once the nanoparticle goes inside the animal model or the human model, so the protein will come and absorb on the surface, protein corona.
Very
So
good.
how we
Yeah, I see your point. And this is what actually before I did animal studies, I wanted to make sure I overcome that, right? So
Right.
you have to be very careful in that case. So what I did was I actually here, if I remember correct, yeah. So what I'm doing here is with the nanoparticles, I actually have a peg which is almost the same size as the RGD peptide. So they are forming a brush combination instead of a mushroom. You see my point? On the surface of the gold. And when you use small nanoparticles, but that allows you actually receptors, basically if you use a small nanoparticle, it actually, because of the surface curvature, it opens up so that receptors can actually access your RGD peptide, right? But when you have to make sure actually you cover the surface with PEG and RGD fully. Otherwise, the opsonine protein in the blood is going to bind to your nanoparticles. Then the circulation time is much less, right? So that's why when we did the animal studies, we see 10 to 12% of the nanoparticle ended up in the tumor because it has a higher circulation. So you can see that. So that's the reason behind. So that exactly you need to cover it up very well. So it depends on the size of the PEG, size of the peptide they use here. It also very much affected. Yeah.
And there is another question like whether the radiation will target only the tumor cells or neighboring cells also, the healthy cells also.
It does. That's where the side effects come from, right? That's where the side effects come from. That's why when I talked to you about this one, I said, this is the tumor control probability with radiation, right? This is the complication to the normal tissue. So what you want to do is this solid curve, you have to push it to the, basically you have to push it to the right. So you have this one here, this curve to the right. So with radiosensitizers, you can push it to the right so that with the 80% control here, you can see that normal tissue damage is much less. But of course, you can't avoid it. You can minimize it. That's the idea.
So there is another question from the student that does the PEG and RGD functionalization not increase the size of gold nanoparticle, decreasing its cellular uptake?
Very good, very good. So the size of the PEG I used was 2K, 2000 Dalton, and the size of the RGD was 1760 Dalton. So they are not very big, right? They are very small. So if you read my recent papers, you will see it's the size of the nanoparticle didn't change very much, but it changes the charge. So if you get gold, it is negatively charged. When you put PEG, it becomes size reduce. So the charge reduces and the RGD is positively charged, right? So at the end of the day, it receives some of the charge, but it is still can survive actually. Yeah, but it didn't change the size very much. Yeah, that to answer that question.
Okay, and how about the stability of the gold nanoparticle for the, how, like in animal studies, like how many days it was stable, or how it is excreted through the routine normal mechanism, or how it is excreted?
Excreted from the body?
The animal model, yeah, from the body.
Okay, so what I noticed was we didn't look at the animal models for a longer time, which I will do at some point. I'm going to, but my point is this: gold has been we have been using gold for centuries, right? For arthritis and so many things. And the idea here is that the gold is not toxic because we did some blood toxicity and things like that with the animal studies. You can measure the toxicity levels. It was not toxic, and the radiation is local, right? So you are not radiating liver, spleen, and other organs. You are just targeting the tumor. Even if the gold is in the other organs, it should not affect you because radiation is targeted to a particular location. So I believe, like I saw some sort of leakage of the nanoparticle from liver and kidney. but I didn't look at it for a long time. But those are some of the things I may look into. And I talked to one of the researchers recently. He said that if we use 5 nanometer gold, less than 10 nanometer, basically, you can actually excrete it through the kidneys easily after the process, rather than getting them into the liver actually. So there are some options available. Yeah.
Thank you. I think that was the last question. So thank you very much, Professor Devika, for your wonderful lecture and answering all the questions patiently.
Thank you.
And
so I
now
will
we can,
stop sharing.
yeah. So now we'll move to the next speaker.
Is that you?
Yeah, Professor Sri Shivakumar. Yeah. So could you share the slides and you can unmute your mic?
Still it is, yes, now I'm sharing the slide.
Yeah, you are audible and your slides are visible. You can
Is it
make
visible
it
now?
Yes, you can make it. Yeah. Yeah. So very good morning, all of you. Now I would introduce the next speaker, Professor Sri Shivakumar. He is currently a professor in the Department of Chemical Engineering, IIT Kanpur. He also serves as associated faculty in Material Science program and coordinator of the Center of Nanoscience and Advanced Imaging Center. He obtained his BSc Tech and MSc Tech from University Institute of Chemical Technology, ICT Mumbai. After his BSc Tech, he spent a couple of years in the industry. Thereafter, he obtained his PhD from University of Victoria in 2006. Dr. Shivakumar carried out postdoctoral research at University of Melbourne. He has received several awards, including the Professor CNR Best Paper Award, Raj and Neera Singh Chair Professor, and Senate Appreciation for the Best Teaching. Dr. Shivakumar's research focus on development of functional nanomaterials for biological and environmental application. He has authored more than 70 publications, 4 book chapters, 11 patents, and 1 edited book. He has obtained 13 grants worth of 13 crores from the government agency, as well as from the industry. Furthermore, he has graduated 12 PhD students and currently supervising more than 14 PhD students. Professor Sri Shivakumar works on synthesis of functional nanomaterials for catalysis and biomedical application. His works on catalysis include both chemical and electrocatalysis. In regard to biomedical research, Professor Shivakumar work ranges from drug delivery to tissue engineering. Now I request Professor Shivakumar to share his lecture and deliver the lecture.
Yeah.
Thank you, Professor.
Yeah. Thank you very much, Dr. Gopinath. And as soon as I see your invitation that I saw University of Victoria, I did not even hesitate to think about it. I immediately accepted your invitation. And I also thank to you for giving this opportunity to present some of our work. In another 40 minutes, I am going to present the work based on the in vitro skin equivalents inspired from the nature. So the skin, there are various skins which is available in the world. The skin which is we live in, for example, flat forms, that is the having the moist skin. And if you see molluscus and then it's having the shell, chameleon, which is having the camouflage skin, and rhinoceros, if you see, it is having a thick skin. And for absolute, okay, it is in the humans, we have a varied skin, which is having the various skin colors, which is there. That means that the skin tissue is distinct across the species. So if you see that, right, the skin, so which I'm going to just introduce the physiological properties of the skin. Human body, that is the which is considered the largest organ is the skin, and it comprises of 16% of the total body weight, and it also expands to fit the body, and it serves as the waterproof. The important one is that the primary uniqueness is that it is the mostly the hairless, and it also protects the body from the various things. One of the things is from the physical assault. And apart from that, okay, if you see the human evolution, that I'm putting a question is that is the human skin is still evolving. So if you see here, that it's a biochemical skin is a biochemical factory because it secretes and it generates lot of proteins and other biomolecules, and it's also a canvas for a self-expression. And though our brain, it is a kind of a serves as it's not totally serves as the brain, but it is, I would say that it is a kind of a brain outside the body. And the average thickness is 1 to 2 millimeter, and the pH is 4 to 5.6, and we have a very good property. The skin has a very good property of the skin renewability. So based on all these things, which is the is somehow okay. It is my screen is frozen. Yes, and now it is started. So skin, it is, if you see, it's a very organizing model. So let us go one by one. If you see the skin, if I do the zooming at the skin, we see some kind of a pattern which is there in the skin. Further zoom, we see some kind of a protrusion which is there, and those things which is having a different layers. What are those layers? These layers which is having three main layers which have been present. One is the epidermis. The epidermis which is having the hairs, as well as which is having the pores, all these things which have been presented, that is the outermost layer. The next layer is the dermis, that's the vascular, which is having the hair follicles, which is having the oil gland, blood vessels, nerves, so all these things which are being present in the dermis layer. The third layer, that is the hypodermis, that is made up of the mainly the fatty or subcutaneous tissues. So in my lecture, I am going to talk about the first two, the epidermis and the dermis. So if we see the cell types which have been present in the skin, there are three kinds of one. One is the epidermis layer. It consists of the keratinocytes, that's the epidermal skin cells. Melanocytes, but that is the one which gives the pigment to the cells. Langerhans cells, that is the immune, and then the Merkel cells. The dermis layer, which has the fibroblasts, immune cells, and the vascular smooth cells. Finally, the hypodermis, that is mainly made up of the adipocytes, that is the fat cells. So apart from that, the important component which is called as the extracellular matrix, which is having the collagen, proteoglycans, and fibronectin biomolecules. So this is the non-cellular component which is present within all tissues and organs, the extracellular matrix. That is the largest component of the cell. It also facilitates the properties of the skins, which has the elasticity, tensile strength, and compressibility. ECM also regulates different cell functions. For example, cell addition. It also lubricates the cells, and it provides a transport system for different nutrients and waste product. So if you see it overall, skin is a very well organized model. It does many jobs. So now the question arises that why do we need a skin replacement? Why do we need an artificial skin? So whenever the human being which is subject to the skin injury, so there are different conventional treatment. So for different reasons to have a skin injury, because of the accidents, because of the diabetic condition, because of the fire accidents or anything, the skin can get injured. So the gold standard is the autograft. That means that the same person which can get the transfer from one part of the body to the next one. The next one is the allograft, from another person to the injured person, the graft which can be done. The third one is the xenograft, that is from the animal model, entirely different species to the injured person. So now the fourth one, the one which is I'm going to discuss in this talk, that is the engineered graft. So that the place the tissue engineering is going to play a major role, which are all made up of from the different scaffold. So now that is the one which is I was talking about, skin replacement. Another one, why do we need a in vitro artificial skin? today morning, the first lecture by Professor Stefani, she has already explained about the drug screening. So here our focus is mainly on to develop the skin models for the drug screening applications. So when we see the drug screening application, so now what is the current practice? It is that we use the cadavers or the explants for the screening of different drugs. So because that is having the improved characterization of the skins and other properties which is mimicking the human being. But what is the challenge? The challenge is that is the ethics as well as there are full ban usage of animals in the scientific research that is implemented already in several European countries, as well as it may come soon in our country, India. So apart from that, there is the challenge, which is that it can have an inherent skin variability if we use the animal model, and they have a different metabolism, and sometimes it can give a inconsistency result. What does it mean? That whatever the one which is getting successful does not necessarily that it may be applicable to the human being. So those are all the challenges, but there are advantages also which are being present because you are testing it in the live system. So now to circumvent these issues, especially for the drug screening application, you cannot sacrifice many animals. That means that that's a ban, which is a restriction, which is there. So to circumvent this one, whether our objective is to develop a in vitro skin model for the drug screening application. So for that one, which is we use the tissue engineering. What is tissue engineering? Tissue engineering, which is having a mainly consists of a scaffold, and which is having the it can made up of different polymers or different material that nature from the nature also it can be used, and it uses several biological factors to culture the cells into functional tissue. So that is called as the tissue engineering, which is having application in many muscles, nerve, different liver, bone, and all these things which is having the application. So now to circumvent our approach, which is having three different ways. One is that to cross the plant and animal kingdom, that's which is we call them plant animal cross kingdom approach. In this approach, which is we use the plant leaves as one of the scaffold. The second one, the paper, the day-to-day paper, which is we use to as a scaffold for the epidermal efferent. Third one, we use the electrospinning, that is made by the cellulose acetate coated with the algal polysaccharide for the dermal skin substitute. Three different approaches which is we tried, and then we got some impressive result. That's what which is I'm going to discuss it in the coming slides. The first one, the plant animal cross kingdom approach. What I mean here is that if you see our native skin, if you closely look at it, it is having the image which is I am showing it here. That is the 3D construction of the cell, which is having the red one that is indicates the nerve fibers, the green which is indicates the elastic fibers. This is the 3D construction. And if you see the cross-sectional view, which is having the nerve fibers, blood vessels, all these things which are being present in our native skin. If you see the plant, especially the one which is I am showing it here is the bougainvillea plant, that's a paper flower. That is also which is having the prominent vasculature, which is somewhat similar to our native skin. So our approach to exploit this bougainvillea paper flower as a scaffold. So when we have this idea, we have gone through the literature and we find some literature already available to use the plants or the plant-based products for the cell tissue engineering or the cell culture. The first report which is being found, that is people who have used the apple, the center part of the apple, which has been used as a scaffold. They have decellularized it, and then they have grown the HeLa cell on the scaffold, and they find it is they have got a reasonable growth on the Apple thing, right? So what? But in order to do that one, what they have done is that they have done the decellularized couple with the collagen hydrogel, which is they have coated with that one. Because of the collagen coating, they have got the improved cell addition and proliferation, and it is also affordable and easy to use as a biomaterial. The next approach, which has been published in 2017, they have used this spinach leaf as the scaffold. They have done the decellularization, but they have to do the biofunctionalization. That biofunctionalization has been done either by the mineral coating or with the dopamine RGD coating, RGD peptide. The previous speaker already talked about this RGD. So these two approaches, apart from the some other approach which is available because of the time, which is I'm not saying. So both approaches, what does it suggest that we cannot use the decellularized bract or the plant thing cannot be used directly. It needs some kind of a coating to adhere into the cells. But our approach is slightly different from this one. So our approach to use the bougainvillea flower, that is which is the family which is the Nyctaginaceae. That is the family which is there. The origin of the country is the South America, and it is bloomed throughout the year. And they are bushes and spread in vines and small trees. And in our campus, we can find a lot of this kind of a paper flower spread in our campus. So how do we choose this one? We have chosen many plants, and then we have done the preliminary studies and details decellularization, and then find that it does not, many of the one except this one does not have the mechanical strength. So what we did is that the real approach, which is I'm explaining, we have taken the bougainvillea flower, put it in the surfactant. In the day one, which is we see the decrease in the pink color. Okay, so that means it started losing its color, and it becomes towards the translucent. The day two, which is if we see here, is that it becomes completely translucent, but we see we also see the prominent vascularized venation, but it's not become as a transparent. The day three, we find it has become entirely transparent and the decellularized, and which is having a reasonable mechanical strength for the cells to grow. This mechanical strength is at par with the skin. So the completely decellularized fats obtained after the 3 days. So now we want to confirm by using the confocal microscope. So whether the native bract, which is having the fluorescence or not. This is a native bract which is having the green and the red fluorescence, but the decellularized one does not showing the fluorescence there. And similarly, which is we have done it with the DAPI staining. So this is the decellularized one, does not show any DAPI fluorescence that is arising from the nucleus. And we also calculated the DNA content that is having the lesser than 25 nanogram per milligram of tissue, which is very, very less. That means the decellularized bracts were devoid of native pigments and nucleus, that is the cell. So now we have done the further characterization with the scanning electron microscope. This is the fresh bract, which is having the protrusion, which is you are seeing it. This is the decellularized one here. So from the one we found that decellularized one, which is having the lesser protrusion, but which is having, which shows some kind of a roughness. Whether that roughness is in the nano size or whether it's in micro size, to find out, we have done the atomic force microscope. From the atomic force microscope, what we found is that this is the one which is we found. What we found is that it is having the height something around in the order of 200 nanometer. So that suggests that this bract having the nano architecture, which is very, very important one for the cell growth. There are a lot of literature that the nano roughness promotes the cell growth. Once we did it, we want to find out the contact angle measurement, because the cell growth is depends upon the contact angle. The native BRAC that is having 190, and for the decellularized fat it is having 127. It's a small increment in the hydrophobicity. So we have done the growth of the fibroblast on the decellularized BRAC. We have done the scanning electron microscope, find many cells which have been grown, and we have also done the MTTSI for the growth of the cells. At the 6th day, we found there's a lot of cells which have been grown onto this one. That means the cellular addition and proliferation will significantly increase after the incorporation of without after the removal of the plant cell, but we did not incorporate any cell addition moiety. This is mainly due to the nano reference. So now we have done the detailed study. The detailed study involves the live cell staining and then the nuclear staining DAPI, as well as different immunofluorescence studies which is we have done it. So this is the one which is after the 6 hours, 24 hours and 48 hours, 4-day, 15-day and 30th day. So this clearly suggests that which is we have done the live cell, that is the fluorescent diacetate, all show the live cells, and the propylium iodide that shows the dead cells. So that clearly suggests that till 30th day we did not observe any dead cell. That means that high cellular viability throughout the 30 days. So NIH3T cells, the one which is we have used, that is the dermal equivalent. Then we have done the apoptosis assay, that is the one, again 6 hours to 30 days. What we found is that there is no significant apoptosis was observed throughout the 10 days. And if you clearly see that there's lot of cell addition which is happening in this one. So that is through on the sides of the vein, wherever the veins which has been present, that it is going through. The cells are growing in that direction. The next one, we have done the FITC, phelloid and DAPI staining to understand how much the cell growth which is happening, whether it is forming like a sheet or a monolayer or multi-layers, or for that one, again from 6 hours to 30 days. If you carefully see this one, that yes, the 30 days, it's completely covered and it is forming like a sheet. So there is a formation of the sheet after 15 days. So it's having after 15 days, it is forming as a cell sheet, and we have find some tight junctions and everything. That is, I will come to that part in the later stage. The next cell which we have done is the hacket, whether this scaffold can be used as the epidermal equivalent. So I wish to note one point. This BRAC is used, it is the naturally 3D printed model, naturally 3D printed scaffold. So again for the hacket also we have done from 6 hours to 30 days. Even after the 4th day, we find the good sheet formation which is happening there. When compared to the dermal equivalent, this one which is showing the more and more cell growth when compared to the dermal equivalent there. So we believe that can be due to the nature of the hydrophobicity, because epidermal equivalent which is needs a more hydrophobicity, because outside of the skin is more hydrophobic than the inside. So this is after the 4 days. So now we want to know whether this kind of an sheet formation, whether it is mimicking the dermal as well as the epidermal. For that one, which is we have validated with the different biomarkers. So if we carefully see here that this is the hacket, this is the NIH3T3 cells. So which is when we have only the hacket cells, there are certain proteins which is less expressed. When it is forming on the cultures on the BRAC, the proteins are over expressed. That means the keratin 1, keratin 10, keratin 16, as well as the integrin 6. All this one which has been more expressed when compared to the cells which has been grown on the cell culture. So that clearly suggests that this one, it is the kind of a mimicking the the dermal equivalent. For the NAH3T cells, which is epidermal one, NAH3T cells again the collagen 3 and the integrin, which is more expressed. So from again, we have done the immunofluorescence studies for the epidermal equivalent. So we have done the staining of this antibodies thing. So this clearly suggests that this has been expressed, the promoted expression of the epidermal differentiation markers that have been clearly shown from this confocal laser microscope. Similarly, we have done for the dermal equivalent for the collagen 3 and collagen 4. So that is overexpressed when on the BRAC, which is having the NAH3T cells. So again, this also promoted the expression of the dermal protein markers. So all this one will suggest that this BRAC, which is having after the decellularization having the nano roughness, promotes the either the dermal as well as the it can be used as the epidermal equivalent. From this this part which is I summarize that we utilize the naturally bioprinted decellularized fluoride BRAC as a scaffold, and it also can develop the simple, robust, and cost-effective, and it can be a scalable process. And so native ECM of the BRAC, which has been preserved after the decellularization, that's a very, very important one. We did not use any surface functionalization, and this can be used closely resembles for the skin microenvironment. This one, which is we are under the further studies of the drug screen, different drugs and everything under the screening of those things which is processed, as well as some of the nanoparticle diffusion, how it is happening, and the in vivo studies are in the progress. The next approach is the paper-based. So why don't we use the paper as a scaffold for the epidermal equivalent? So we know the Asian invention, right? Okay, the paper and the modern paper that has been invented by the Chinese. So can we use this paper as a scaffold, especially for the epidermal equivalent? Again, we went through the literature. It has been already exploited. For the, especially for the 3D cell culture, it has been exploited. So again, if you see paper, it needs a modification because it's very, very hydrophobic. So it hydrophilic. So it needs some modification. Otherwise, okay, cell addition will not happen and it will not be suitable for cell culture. So they have done the modification either with the wax or with the PDMS polymer, which is that. So which has been used. So this has been used for the 3D cell culture one, which is it, but not being exploited for the skin equivalent. And it also needs for the skin equivalent, you need a similar kind of an architecture which is needed when compared to the approaches available in the literature. So how which is we thought the motivation here is, so if you see the physiology of the human epidermis, so it is having a very strong and flexible, compact and self-repairing barrier. Is the waterproof. Again, it protects our body from the dehydration and unwanted substances. The important one is that highly stratified and having the fibrous ECM. And it is also having the presence of the nano or the micro tomography. And important one, I just want you to emphasize here that from the top to bottom, if you hear that, or from the bottom to top, it is having the more increasing the hydrophobicity. What does it mean? The top layer is more hydrophobic, and if you go down from the top, hydrophobicity it is decreasing. But and it's also having the decreasing the compactness. So when we develop the scaffold, it should have a similar kind of a property so that which is we can grow the cells in this parameter with this architecture. So for that one, so we propose the following epidermal scaffold. We take the paper, which is having the microfibrous matrix, but it's highly hydrophobic. Because of that one, I already mentioned, which is having the poor cell addition, and which is it's also a loosely bound and thick. So in order to introduce that the gradient in the hydrophobicity, we follow the following approach. The first one, what we did is that we coated one side of the paper with the polycaprolactone nanofibers, which is made by the electrospinning method. So this polycaprolactone is having a very high hydrophobicity, dense and thin, and is having the nanofibrous matrix. The the other side of the paper, one side is coated with the polycaprol, the other side we coated with the polydimethoxysiloxane nanoparticle, not as the polymer. So it is as the polymer nanoparticle which has been coated. So the reason behind here is that it induces the nanotopography, and it also it provides the anchoring sites for the cell addition, and it introduces the moderate hydrophobicity. So we change the surface of the PDMS such a way that it is not having hydrophobic like caprolactone, but it is having the lesser hydrophobicity. That means that based on this one, which is we can create the increasing the hydrophobicity from the moderate hydrophobicity to the highly hydrophobic, as well as which is we can have a decreasing the compactness. So with this one, the fabrication strategy here is that we take the paper using the electrospinning, we electrospun the caprolactone nanofibers on the paper, and we take that PCL coated nanofibers paper, putting it onto the PDMS nanoparticle particles disposed in the PBS. Because of the nanoparticle which has been deposited onto the other side of this one, because of the hydrophobicity, the top part which is not exposed, only the bottom part which has been exposed to the nanoparticles. So nanoparticle which has been made by the nano emulsion method, which is I am not explaining this one there, which has been published in the paper already there. So this is the some image of the nanoparticle. These are all the sizes in the order of 50 to 60 nanometer. So once we have it, and then we have characterized with the contact angle measurement there. So this is the only the free paper, the contact angle 0, because the total nano water which has been spread. This is the nanofiber, this is the filter paper with the nanoparticle coated with the PDMS that shows the hydrophobicity is the 125. This is with the PCL with the nanofiber as well as PCL nanofiber with the filter paper as well as the nanoparticle. This is the filter paper, that means the PCL nanoparticle site, and then this is the nanoparticle site, and this is the PCL site. You see the increase in the hydrophobicity from 121 to 134. So that creates a kind of a gradient in the hydrophobicity. With this one, we have done the in vitro studies. What we found again, which is we have taken the macaque cells then, because that's the epidermal equivalent. We gone with 24 hours to 7 days. What we found, it is having the very high cell proliferation thing when compared to the only the filter paper, as well as compared to the nanofibers, only nanofibers. So, and similar to the last studies, we have done the detailed study of the staining Fitzy and Phellodoin, and we found after the 7 days, a lot of cells which have been growth on the nanoparticles and PCL coated filter paper. So that clearly suggests, okay, this can be used for the forming the epidermal layer. So that's the sheet formation which is happening after the 7 days. So again, like previously, we have done the immunofluorescence study. This is a cross section. So different antibodies which is we have taken, and then we have done the studies. What clearly, which is having the clearly studies from the cross-sectional images, the D is the one which is having the PCL as well as the nanoparticle coated on each side of the filter paper, showing the more and more expression of this antibodies in immunoglobulin and cytokeratin 10 and cytokeratin 14 when compared to the control sample. That's the only filter paper or nanoparticle coated, or only the caprolactam nanofiber coated. When we have both the sides, the expression of this one, it is more and more. So that is also which is giving the indication that it promotes the expression of the epidermal differentiation markers. We have done the scanning electron microscope to characterize the nanofibers that, and then we find these are all in the order of the nanofibers, or in the order of 200 nanometer. So this formation, it is composite with the decreasing the compactness. That is the one which is needed for this kind of a scaffold. So again, we have done the energy dispersive X-ray spectroscopy to find out the PCL1 that. So that and the EDEX that clearly suggests that it was all having the nanofibers which has been present there. We have done the in vitro studies for the FESEM for the addition of the cells that has been confirmed after the 7 days. A lot of cells which have been adhering onto the paper. So from this part, I summarize. We fabricated paper-based PCL nanofiber with the electrospinning, and the PDMS nanoparticle modification is absolutely necessary, and that reduces the gradient of the hydrophobicity, that variation in the macro and micro and nano level topography, and this may be used as a epidermal scale macro environment, and further studies with the drug screening as well as the nanoparticle diffusion is under progress. The third approach, we have used the cellulose acetate nanofibers coated with the algal polysaccharide. So again, this has been we propose this can be used as a dermal component. If you see the human skin, as I already mentioned in the starting of the layer, epidermis, dermis, and hypodermis. The dermis is the one which is contain the elastin fibers, fibroblast, as well as the collagen, and which is there. And this also composed of the polysaccharide chains made up of the repeating disaccharide series as and are strongly hydrophilic. So we want to exploit this one for especially for the electro ECM one. So here we want to exploit the carrageenan, that is the polysaccharide material disaccharide one, which is having the two galactose molecule joined together, which has been can be synthesized from the red algae, and it's also popularly used in the food industry for as the emulsifier, stabilizer, and it has been exploited in the as a gelling and thicknessing agent. So it's like highly biocompatible. So what we thought, whether we can exploit the this carrageenan as a scaffold by converting into the or fabricating as a nanofiber. So when we again went through the literature, so this has been exploited in the wound dressing material, and it also promotes the cell migration and the reepitheliation, and it also having the promising effect on the healing of chronic wounds, and it also enhances biomineralization and osteogenic differentiation. The objective here is that, so we want to develop the electrocellular cellular matrix, the mimicking the dermal skin substitute using the carrageenan modified cellulose acetate nanofibers. So why do we want to use cellulose acetate? So the cellulose cellulose, it is the most abundant, biocompatible, biodegradable, and non-immunogenic. So if we incorporate this kappa carrageenan, so it can give you, okay, I have already mentioned that it's having the gelation capability, good, giving a good mechanical strength and similarity with the ECM. So combination of this kappa carrageenan as well as the cellulose can make a scaffold which can mimic the extracellular matrix. So the fabrication strategy, we use the electrospinning. So after the electrospinning, what we did it, we made the cellulose acetate nanofibers, and we just take those fibers, coating it to the kappa carrageenan solution. We have done 1 coating, 2 coating, 3 coating. So different coatings which is we have done it, and we have optimized that number of coatings. We have done the characterization. So nanofibers with the coated with the kappa carrageenan, which is with the SEM studies. So that clearly suggests that the size does not increase much after the kappa carrageenan coating, and the morphology, this nano architecture, did not change much after the coating. So we have done the X-ray EDEX to confirm the presence of the kappa carrageenan, that the EDEX that clearly suggests it is present. And we also done the toluene blue staining of the scaffold, so that also supports the presence of the kappa carrageenan. So we have done with the different coating level 0.1, 0.5, and 1%. That depends upon the different coating levels. So we have done contact angle measurement. So this is the cellulose acetate fiber. So native one that shows the 115. When we coated with the 0.1, 0.5 and 1%, the contact angle considerably decreased. So that is the one which is we needed, that we need the hydrophilicity for the growth of the cells. So that's similar to the damage. We have done the FTIR analysis to confirm the presence or to confirm the presence of the sulfate groups. We have done the in vitro studies. So this is with the native one, with the coating, without the coating, and 0.1%, 0.5 and 1%. That after the 7 days, the growth of the NIH3T cell, that is the dermal equivalent cell lines. So that shows very good growth after the 7 days, the high cellular addition which is happening there. Then the life and dead cell assay, MTT assay, that also supports the growth of the cells. We have done the in vitro studies, that is the fitzicolloid one. So after the after the 7 days, which is we can find a very thick shell of the NIH3T cells, which is forming over the scaffold. We have done the RT-PCR analysis to find out the markers, which is due to the collagen dermal equivalent, collagen 1, collagen 3, and then the taline, and all this one, which clearly suggests overexpressed that it can be used as a dermal equivalent. Again, further confirmed with the immunofluorescence studies with the collagen 3 as well as the collagen 4 and collagen 6. So the collagen 3 and 1, which is you can see it is overexpressed when compared to the uncoated, the control samples are there. Similarly for collagen 4 and collagen 7. So these are the one which has been confirming the expression of the different protein biomarkers which has grown onto the kappa carotene coated fiber matrix. So from this one, I summarize. We successfully modified the cellulose acetate nanofibers with the kappa carotene. So this promotes the growth of the dermal equivalent cell, and the antibody staining, it shows the presence of the dermal protein biomarkers which is responsible for the formation of the skin dermal equivalent. With this one, I acknowledge the students who is involved in this work, again, as well as the Prerna, and our collaborator from Tata Consultancy Services, Dr. Beena Roy. And this project was majorly sponsored by the industry, the Tata Consultancy Services, and a part of that one, it is funded by the Department of Biotechnology. Thank you very much for the patient listening.
Thank you, Professor Sri Shivakumar. It was really a wonderful talk, and there are a lot of interesting examples also. So there are some questions from the participants, which I'll read it out on behalf of them. So how closely do the in vitro skin models mimic the animal skin models?
See, that is under the progress, which is we have done, see, due to the COVID situation. So we started the work and then it stopped. It is now, which is we again started the work, which is we are going to evaluate these things with the animal models. Okay. So, but I did not mention certain things because that's an industrial component which has been involved there. We found that it can be used as a drug screening model. That is the one which is the major focus, but now we are extending whether it can be used as a skin equivalent for the tissue engineering applications.
So another question is, which is the best method among the three methods?
Okay, the first method, the Bougainvillea one, that is one which is we prefer because that is does not preliminary studies does not induce any much toxicity or anything which is there, and which is having the mimicking of the nerves and everything, so that blood transfusions and all these things which is possible with that one. We prefer the BRACT model.
Okay, and the next question is that deceleration surfactant use have any effect on removing the lignin present on the surface of the BRACT?
It is not much only.
Okay.
Yeah, we have not quantified it, but I believe that.
Okay, another question is, in one of the slide you showed that some research group, they have used apple as the scaffold.
Yeah.
So is there any specific reason for selecting that fruit as a scaffold, or?
I think they might have tried a different one, because that is again the 3D naturally available 3D model, right? That's the reason they have used it.
Okay, okay.
Because it's having a good mechanical strength. Okay, that may be the reason.
Okay. And if you are printing if you are making that artificial skin, like how it can be connected to the blood vessels, like if you want to implant to the patient, so the blood veins and connectivity, and how it can closely match with the body tissue.
See, okay, that is needs a lot of further studies, which is needed. That so once we implant this kind of models into the animal one in vivo studies, we will come to know that. But these are all made up of with the biocompatible one, so it can be easily degraded in the body, and then the cells can.
Okay, okay. So yeah, so there is no other questions. Then let us thank Professor Sri Shivakumar for his wonderful talk, and thank you, Professor Sri Shivakumar, for immediate acceptance of our invitation and also delivering a wonderful lecture and be with us on this day. Thank you very much.
Thank you so much. Thank you so much. And now I will unshare the slides.
Thank you.
It is my pleasure to introduce Dr. Gopinath. Dr. Gopinath is an associate professor in the Department of Biotechnology at Indian Institute of Technology, Roorkee, India. He received his BSc in microbiology and MSc degree in biotechnology from Bharati Darsan University, India. He earned his PhD in biotechnology from Indian Institute of Technology, India. He did his postdoctoral research at University of Rochester Medical Center, New York, United States. Currently, his research group in Nano-Biotechnology Laboratory at IIT Roorkee is working on the development of polymer-based nanocarriers for delivery of various therapeutic agents. His group is also exploring new avenues for synthesizing novel biocompatible imaging agents. At present, he has more than 111 research publications in the area of nanobiotechnology in high-impact journals. He has filed 10 patents and done 1 technology transfer. He has also published 8 books and 13 book chapters. He has been a reviewer and an editorial board member for many international journals. He has received numerous prestigious awards for his scientific contributions in the area of biomedical nanotechnology. So I would welcome Dr. Gopinath to give his talk. Thank you.
Yeah, sure. So the next 40 minutes, I'll be talking about cancer nanotheranostics. So how we can use nanomaterials for cancer diagnosis and cancer therapy. Okay. So before that, I'll give a brief introduction about the nanoparticle. So what is nanoparticle? As most of you know, that the nanoparticle is in the range of any material which is in the range of 1 to 100 nanometer is called as nanoparticle or nanomaterial. Okay. And the word nano means it's a Greek word. It means dwarf or small. And most of you know that nanometer means is 1 billionth of a meter. That is a 10 power minus 9 meter. Okay. So let us see a simple example to understand the scale difference. So if you see the whole population of India, that is 1 billion, that is 100 crore. In other words, it is 10 power 9. So when you compare to the whole population of India, you and me are nano size. So that much small is the nano. And the next simple example is it is equal to 1 rupee in 100 crore rupee. So that much small is the nano size. So why small is good? So why you want small particle? What is the advantage of this small particle? So when you have the small nano size particle, the surface area to volume ratio will be higher. So when the size goes down, the surface area to volume ratio will be higher. For example, here you can see here, when you have the 7 shell, the surface atom is only 35. And when you have a 1 shell, the surface atom is 92. So when you have the smaller material, then it can be very, very faster, and it can be lighter, and it can be more energy efficient. So that is the advantage when you use the smaller size particle. So let us understand how the surface area to volume ratio will be increased when the size goes down to nano range. So for example, this is a 2 mm box. The same 2 mm box you are cutting into small pieces, like equal to 1 mm. Okay, so then when you apply the formula, the surface area, and when you calculate, the surface area is 48 for this 1 mm and 24 for this, and the volume will be the same. So when you calculate the ratio of surface area to volume ratio, it will be higher for the nanomaterial when compared to the bulk material. So, and again, this nanomaterial will be having more reaction, more reactivity, more catalytic activity. How it is possible? If you see that when you break the bigger size material into the nano size, the hidden surfaces are exposed. Okay, so the hidden surfaces are exposed, and that leads to the better catalyst and also the greater proportion of the material to expose for the potential reaction. The simple example or analogy to understand this concept is, for example, you are having an aspirin tablet. When you have an aspirin tablet, you put into water. It will take at least 50 seconds to dissolve the complete tablet. And the same tablet, you crush it into powder and put it into water. It will take only 10 seconds to dissolve into the water. So when you make the bulk into the smallest size, though it will have more catalytic or reactivity, okay, because the hidden surfaces are exposed. And whether the nanotechnology is new or not. So again, similar to the biotechnology, the nanotechnology is not a new. For example, if you see the old churches, so these are the beautiful paintings. These beautiful paintings are made with the gold nanoparticles, gold and silver nanoparticles. So those days they don't have any commercial color paints. So they used gold and silver to make these beautiful paintings. And with respect to the sunlight and transmitted and reflected light, it will give a different kind of color with respect to size and with respect to shape of the various nanomaterials. And also in those days, they have also used this Lycurgus cup, which is in the 4th century Roman glass cage. Okay, so they used silver and gold to coat this the cup, okay, which the king and queen use it. So those days they don't have the graphics, so they use gold and silver, and when the light shines from the inside or outside, it will appear red and green color. And again, it is not new to the Indian traditional medicine like the basmas and other things, which also have the metal colloids into that. Okay. So it is not new to the Indian traditional medicine. So let us see how to make the nanostructures. There are two approaches to make the nanostructure. The first approach is top-down approach. So top-down approach means building something from the bigger material and making into a smaller structure. It is similar to making a statue from the rock, from top to bottom. That is a big size to the small size. And next approach is bottom-up approach. So bottom-up approach is like from a very, very small size, you are reaching to the nano size. For example, you can use atomic scales and make the nano scale. So the simplest example is using the brick and constructing the building. Okay. And the simple example or the biological example is like if you take the nucleotide or amino acid, these are in the range of atomic scale. So these amino acids combine and form the nanoscale protein. And similarly, these nucleotides combine and form the nanoscale nucleic acid, DNA or RNA. So that is the example for the bottom-up approach. And the next important thing is the nanoparticle, we can make different shapes and different size. And also we can use a different kind of material. We can use the polymer, or we can use the metal, or we can use the hybrid material. So that is possible. And very important thing is the surface charge. Okay. So if you see that most of the biological material, it has a surface charge of negative charge. For example, if you take a DNA or if you take a cells, it will be having a negative charge. And when you have the positive charge nanoparticle, so it can easily bind to the negative charge cells. So if we have a positive charge nanoparticle, it can go and bind through electrostatic interaction. So that is one approach. Then another approach is we can also add a targeting ligand, as explained by Professor Devika. So we can add an antibody or you can add a peptide which is specific for the cancer. Then it can go and specifically bind to the cancer cell. And we can also add a group called polyethylene glycol. So that will enhance the circulation time. So otherwise, these nanoparticles can be removed easily. So when you add the polyethylene glycol, it will increase the circulation time. So let us see that what is the difference between nanoscience and nanotechnology. So if you are making a fluorescent nanoparticle, that is quantum dots in your lab, that is nanoscience. And when you use the same quantum dots and make a quantum dot based LED TV, that is a technology. And let us see the difference between nanobiotechnology and bio-nanotechnology. So nanobiotechnology means when you make a nanoparticles for biological applications. For example, you are making a nanoparticle for cancer diagnosis or cancer therapy. So that is nanobiotechnology. And the bio-nanotechnology means it's understanding the biological nanostructure. For example, DNA and protein, these are in the nanoscale. And when you understand these biological nanostructures for application, for example, we can use the DNA as a construction material for making the DNA-based nano cages for drug delivery or for sensing application. So that is called as bio-nanotechnology. So understanding the biological nanostructures for various applications. So let us move on to this cancer nanotechnology. Before cancer nanotechnology, so I'll briefly talk about what is the difference between cancer and tumor. So most of you know that tumor is localized. Okay, so that is a benign, so it does not kill the host, and this tumor can be removed by surgery. But when it became malignant, that means the tumor grows indefinitely and it will start spreading from one location to other location. That process is called as metastasis, and it spread throughout the body and it start killing the host. So that is the cancer. Okay, and if you see the types of cancer, the 90% of cancer is carcinoma. Okay, mainly the breast cancer, skin, and the digestive tract and respiratory system. Okay, and if you take the male population, so in the male population, the lung cancer is dominant, and in the female population, and the breast cancer is dominant. So if you see the reason, what is the reason for this cancer? It's maybe due to the genetic factors. For example, some of the breast cancers are due to the genetic factors. If the grandmother is having the breast cancer, there's a high chance for the mother or the granddaughter to get the breast cancer. Okay, and some of the environmental factors like UV radiations or chemicals, when you're exposed to the particular chemicals for more time or a more amount of time, so that may also mutate your DNA and it may also leads to cancer. So how the normal cell will become a cancer cell? For example, each cell will be having a proto-oncogenes. That means that is potential to become a cancer cell. So when the proto-oncogenes become oncogenes, then the normal cell will become a cancer cell. So in the cell, we'll be having a growth promoting proto-oncogenes and growth restricting tumor suppressor genes. And these two should be properly balanced. So it is similar to the person will be having a good character and bad character. So it will be properly balanced. Suppose if there is an imbalance, if the bad character will dominate. Okay, the same thing happens. If there is a alteration in the tumor suppressor genes, or if there is a conversion of proto-oncogenes to oncogenes, then it leads to the uncontrolled cell growth. So that leads to the cancer. Okay. And according to recent research, it shows that at least once a day, your immune system is killing a cell which could become a cancer cell. That means every day, everybody is having at least some cells trying to become a cancer cell. Our immune system is going and destroying it. Okay. So from this animation, you can check it out. So you can see here, this is a cancer cell, and your immune cells coming and attacking the cancer cell. Okay. So let us see how we can use these nanomaterials for cancer diagnosis. So we can use this quantum dot. These quantum dots are the semiconductor nanomaterials, nanocrystals, and with respect to size, it will give a different fluorescence. So we can use these quantum dots for various imaging and diagnostic application. But one drawback of the quantum dot is, so it is made up of heavy metals, and again it's a water solubility. To increase the water solubility, what we can do is we can do the organic coating. When you do the organic coating, the water solubility will be increased. And again, to improve the brightness, we can add an inorganic shell. And for example, if you want to target these quantum dots specifically to the cancer cell, we can add the biomolecules. We can add an antibody specific for cancer, or we can add any nucleotides. So that will specifically go and bind to the cancer cell. So how these quantum dots plays an important role in cancer diagnosis. For example, when you compare to this traditional organic dye, so you can see here this quantum dots. So when you add it on the breast cancer, which is having a high level of expression of the breast cancer marker, so both the quantum dots and organic dye, it is giving more or less similar effect when the breast cancer in the advanced stage. So when you take the breast cancer in the early stage, that is where there is a low level of expression of the breast cancer marker. You can see here in the organic dye, there is no signal. There is no fluorescence. So that means the clinicians or the doctor may say there is no fluorescence and there is no marker for the breast cancer. So the person is safe, no breast cancer. But the same person, when you go for diagnosis using the quantum dot, so you can see here this quantum dot is giving the fluorescence. Okay, that means this quantum dot is able to diagnose the disease in the early stage. The quantum dot, which is tagged with a specific antibody, which can go and bind specifically to the cancer cell and give the fluorescence signal. So based on the fluorescence signal, we can say the person is in the early stage of cancer. So when you're able to identify the disease in the early stage, so that will be more easy to cure the disease. And another advantage is when you use the traditional organic dye, there is a high chance for overlapping of the fluorescence. But when you use this quantum dots, with respect to size, it will give a different kind of fluorescence. So there is no overlapping of the fluorescence. That is another advantage. And again, most of the cancer cells will be having it will express multiple types of markers. For example, in a simple term, you can say it's a blue color and purple color and also red color. Okay. So various types of markers or receptors it will express. So we can have a quantum dots of different fluorescence and tag it with antibody and attach to that, so that we can multiplex the diagnosis. Okay, so whether the cancer is due to the which type of marker. For example, if you are getting the blue, green, and red color, all the fluorescence, 3 fluorescence, we can say the person is in the advanced stage of cancer. Okay, or if you are getting only one kind of fluorescence due to one type of marker expression, we can say the person is in the early stage of cancer. So this is the quantum dots, and which we inject into the mouse model. When we inject into the mouse model, it is specifically going and binding to the tumor cells. You can see when compared to the whole body, it is binding only to the tumor location, and only the tumor is glowing. So when the tumor is glowing, it will be also useful for the person who do the surgery to remove the tumor. So when you do the surgery, you have to remove the complete tumor cells. So if you are not removing the complete tumor, there is a high chance for the reoccurrence of the tumor. That is also one of the challenging aspects. So I'll discuss later how we can overcome using the nanomaterials. So there's another approach called chemical nose approach. For example, when you purchase a bag full of apple, and if any apple got rotten, so you don't send the apple bag to chemical lab for checking which apple is rotten. Instead of that, you just smell it and tell which apple is rotten. The same concept here. So we can make a chemical nose by using 3 different polymers, and also we can use 3 different kind of fluorescent nanoparticles. For example, assume this is like a green fluorescent, and this is the red fluorescence, and this is the blue fluorescent. Okay, so these 3 different fluorescent nanoparticles are covered with the polymers. When it is covered with the polymer, the fluorescence will be off. And these polymers are specific for the cancer cell receptors. So when it binds to the cancer cell receptors, the polymer will be removed. So once the polymer is removed, the fluorescence will be on. Okay, for example, if you are injecting this nanoparticle into the normal person, you don't get any fluorescence signal. So if you inject this nanoparticle into the cancer patient, you get the fluorescence. For example, if you are getting only one type of fluorescence, we can say he's in the early stage of cancer. If he's giving all the 3 color fluorescence, we can say he is in the advanced stage of cancer. So that is one approach. And most of you know about this pregnancy test kit, okay, which is available in any medical shop, 30 to 40 rupees. And when you put a drop of urine sample, or when you put a drop of water, you can get a single line. If you get a single line, that means the kit is working. So if you put a drop of urine and the particular person is getting 2 lines, that means the particular person is pregnant. And this pregnancy test kit can be also useful for cancer diagnosis. So if a man take the pregnancy test kit, and if he get positive result, doesn't mean that he is pregnant. Instead, he has a high chance for the testicular cancer. Okay, so if the man has the chance for the high chance for the testicular cancer, he will also secrete the hormone, the HCG, in the urine sample. So that can be useful for diagnosing this testicular cancer using the simple 30 to 40 rupees, the paper-based kit. So in our lab also, we are trying to make such kind of paper-based diagnostic kit for identifying the early stage of cancer. So let us see how we can use the nanomaterial for cancer therapy. So how this nanotechnology can be better than the traditional method. So in the traditional method, you can see here, when you add the drug or when you inject the drug, what happens is the drug don't know which is normal cell or which is cancer cell. So the drug will go and attack the healthy cell as well as the cancer cell. So that is a major drawback or major side effects with the traditional treatment. Okay. So when you use the nanoparticles, what happens is these nanoparticles are specifically target only to the cancer cell. So it will go and attack only the cancer cell, and the healthy cells will be happy. So the healthy cells are protected, it is not getting affected. So that is the advantage of this nanomaterials based cancer treatment. So you can see here this animation. So this is a kind of nanobots. It's called as a nano robot. So we can have this kind of like a soldiers in the body. Whenever any pathogen or whenever we have the cancer cells, it will find it out and it will destroy it immediately. So how to target these nanoparticles? So there are two types of targeting. So the first one is passive targeting. The next one is active targeting. So the passive targeting, in a simple term, it is like sending a letter to Prime Minister of India. So if you want to send a letter to Prime Minister of India, you can put Prime Minister of India, New Delhi. The letter will reach. Okay, you don't have to write any other address. So the same here. So if you want to target the nanoparticle to cancer, so you can make a polymeric nanoparticle in the size range of 200 to 250 nanometer, and which can reach the tumor location. Okay, because the tumor location have a lot of leaky blood vessels. So in the leaky blood vessels, it can go and bind and it can release the drug. That is called as passive targeting. And next one is active targeting. So this active targeting is sending the letter to your close friend. Even though he's your close friend, you have to write the address with the proper street number, house number, and also the pin code and zip code and even the mobile number. So then only it can reach the particular person. So active targeting is to the nanoparticle, you'll be adding specific antibody so that this nanoparticle can go and specifically bind to the cancer cell. So that is the difference between passive targeting and active targeting. So let us see in details. So here the tumor cells need more amount of blood to grow larger than 2 mm size. So when it need more amount of nutrients and growth, what will happen? These tumors will be having their leaky blood vessels. So we can make use of these leaky blood vessels, and when you make a nanoparticle of 200 nanometer size, so it will be trapped in that area. So that effect is called as EPR effect, enhanced permeability and retention effect. Okay. So due to the EPR effect, it will be trapped in the particular tumor location. So you can see here this green color is the tumor location. Okay, and you can see here there are a lot of leaky blood vessels. The blood vessels are not stable. So when compared to the normal blood vessels, so here the normal blood vessels, there is no damage, it is perfect. And in the tumor region, this green color region, you can see here there are a lot of leaky blood vessels. So in the leaky blood vessels, these nanoparticles will be trapped and it will release the drug slowly and it will kill the cancer cell. So this is called as passive targeting. And in case of active targeting, you can add antibody. We can add an antibody or we can add the RGD peptide. This is the arginine, okay, arginine, glycine, and aspartic acid, aspartate. This is a 3 amino acid peptide. So most of the cancers this express, so which can easily go and bind to that. Okay, or we can also add another chemical, that is the folic acid. So most of the breast cancer express receptor for the folic acid. So we can add a folic acid and which can easily go and bind to the folic acid receptor in the breast cancer, and it can release the drug only to the breast cancer, not to the healthy cell. So these are the advantages when you use the active targeting. So let us move on to the research work, what we are doing. So we are working on development of various nanomaterials for various biomedical application. And in my today's talk, I'll be mainly focusing on what are the various nanomaterials we made it for cancer diagnosis and cancer therapy. So this is a first research paper we published 10 years ago. And that time when I started the lab, we are struggling with the minimum lab facilities. So we made the fluorescent nanoparticles with the simple microwave oven, which we use it for heating our coffee or tea. So using the simple microwave oven, we made the fluorescent nanoparticle. So as I told, the quantum dots have several drawbacks because these quantum dots are made up of heavy metals. Okay, and it is not water soluble, and it also have a lot of toxicity. So to overcome that, we can use the carbon dots. So this carbon dots is basically a black material with a low solubility and no fluorescence. But when you make the carbon dots, so when compared to carbon, these carbon dots are highly water soluble and it will give a very bright fluorescence. Okay, And these carbon darts are 0 dimension. It don't have any dimension. It is less than 10 nanometer in size. Okay. And if you, most of you might have seen that, what is that like? When you have the kerosene lamp, there is a lot of black colored things are sitting on the sides of the glass. So that is the carbon nanoparticles. Or if you burn any carbon source, okay, if you burn a paper, the black colored things, that is the carbon. So any carbon source, if you burn, you whatever you get, that is in the range of carbon nanoparticles, that is carbon nanodarts. So let us see the difference between quantum dots and carbon dots. As I told earlier, this is a quantum dots is made up of heavy metals, so it has high toxicity. But the carbon dots are basically carbon source, so it's highly biocompatible and very simple to make. And also we can easily add the functional group. So when you add the functional group like amino group or carboxylic group, it is very easy to add your antibody so that we can easily target it to the specific cells. And it's highly water soluble. So these are the advantages of these carbon dots. So we used chitosan as a carbon source, and we used a polyethylene glycol as a kind of capping agent or the stabilizing agent. And we used sulfuric acid as a dehydrating agent. And we use a simple microwave one, which we use it for heating our coffee or tea. So when you heat it for 2 minutes, and we got the multicolored fluorescent carbon dots. Okay, so this is a very simple protocol which you can also try in your lab, or you can use it as a kind of your undergraduate projects. Okay, so this is a protocol. The details about the protocol is available in this journal, and I also shared the complete protocol. So it is very easy to make. So we can use only 0.2 gram of chitosan and add this much amount of sulfuric water and 4 ml of sulfuric acid, and add PEG and mix it for 15 minutes. Then use the normal domestic microwave one and use it for different cyclic times. So this step is very, very important. So don't keep it for 2 minutes continuously. So when you keep it for 2 minutes continuously, your glass flask may blast. So what you have to do is you have to keep it 20 seconds on and 10 seconds off, and take it out, and again 20 seconds on and 10 seconds off. So you have to repeat the steps until you get this kind of brown color solution. When you get the brown color solution, you can centrifuge. Then what you get is supernatant, that is having the carbon darts. Okay, and we taken these carbon darts, and those days we don't have the cancer biology lab. So what I did is we use a simple bacteria, the E. coli bacteria and Staphylococcus aureus, the gram-positive bacteria. And when we add these carbon darts, and it was giving a multicolor fluorescence. Okay, with respect to the excitation, it was giving a multicolor fluorescence. So we took this work further for cell imaging. So what we did is, in the next work, what we did is we want to make the fluorescent nanoparticle with the positive charge. So we taken the same chitosan, and when you add the polyethylene imine, so it will give this amino group will give the positive charge. It will give the positive charge to the nanoparticle. And we also did the same nanoparticle, prepare the same nanoparticle with the PEG. So this will give the negative charge nanoparticle. And again, here there is a challenge. So we want to confirm the positive charge and negative charge, and with a very, very simple experiment. So what we did is we taken the simple agarose gel electrophoresis. We taken the simple agarose gel, and we made the well in the middle of the gel. And when we loaded this nanoparticle, and when you apply the power supply, this positive charge nanoparticle move towards this negative charge, and this negative charge nanoparticle move towards the positive charge. So with the simple experiment, we are able to identify the charge of this nanoparticle. And when you add it on the cancer cell line, lung cancer, and the normal cell line like baby hamster kidney cell lines, so you can, as expected, this polyethylene, which has the positive charge, so that is having more fluorescence because it has a more electrostatic affinity to the cells. It is binding to the cells, and it's giving more fluorescence. So the next approach is, instead of having this fluorescent nanoparticle separately and the therapeutic material separately, can we combine and make the theranostic nanoparticle? So the theranostic means it's a combination of therapy and diagnostics. Okay. So the single nanoparticle, that is a multifunctional nanoparticle, which will be having therapy as well as the diagnostic function. So we have also published a book in this area. So the next work, what we have done is we have taken this carbon darts, so which is negative charge carbon darts, and we have taken this positive charge metal nanoparticles, okay, metal nanocomposites. So these metal nanoparticles will induce cell death through apoptosis, that is a programmed cell death. And we have added this carbon darts, which is a green color carbon darts. So due to the electrostatic, when you mix these two nanomaterial, it forms this kind of theranostic nanoparticle. Okay, so which can induce the cell death. At the same time, it can also give the fluorescence signal. So when we add it on the cell, we can see here when you add only the carbon darts, the cells are healthy. The cells are not dying. The cells are like spindle shape. Okay, the cells are healthy. And when you add the carbon darts decorated nanocomposites, you can see here with respect to concentration, when you add the high concentration, the cells are undergoing cell death. So when the cells are undergoing cell death, so these elongated cells will become like a rounded cells. Okay, so that you can easily observe the cells become rounded and the cells are undergoing cell death. And when you taken out the cells and when we see under the scanning electron microscope, so you can see here, so the normal cells, the untreated cells, these are like a star shape or the spindle shape. And when you treat with the nanoparticle, it is undergoing apoptosis, so the cells become rounded. Okay. And most of the metal nanoparticle induce apoptosis or cell death through reactive oxygen species, ROS, reactive oxygen species. So to confirm that, we use a very simple assay that is called as DCFH-TA. So this is a very low cost dye. It's like 3,000 rupees. So when you use this dye and when you add it on the cells, which is undergoing the cell death, when you add it on the dye, when you add this dye to the cells, what happens is this non-fluorescent dye will be converted into fluorescent in presence of reactive oxygen species. So if we have more amount of reactive oxygen species, you'll get more fluorescence. Okay, so when you add this dye to the normal cells, you don't get any fluorescence. When you add this dye to the cells which is undergoing the cell death due to reactive oxygen species, then you get more fluorescence. So that you can check it using the fluorescent microscopy or fluorescent spectroscopy, and also you can do it using the flow cytometry. So you can see here this is a control cells, normal cells. So there is R2 is a non-fluorescent. So in the R2, in the control cell, it is almost nil. Okay, and you can see here with the treated cells, you can see here the amount of fluorescent cells are increasing. That means with respect to reactive oxygen species, the fluorescence is increased. And this work was highlighted as the cover page of the Journal of Metal Chemistry B. And the next work, instead of using the metal nanoparticle, we want to use the polymer. So here we have selected a polymer called a dendrimer. So the dendrimer is like a tree-like structure. It is in a Greek word. Dendron means it is like a tree-like structure. So this polymer will be having a tree-like structure. So we use the dendrimer, and in the dendrimer we loaded an anti-cancer drug called epirubicin. So the advantage of using this drug epirubicin is, so the epirubicin itself have the red color fluorescence. So the drug itself have the red color fluorescence, and on the top of that polymer we have added carbon darts. So this carbon dart is, the carbon dart will give green color and the drug will give the red color fluorescence. Okay, so using that we made a theranostic nanoparticle, and this drug is specifically will go to nucleus and kill the cells. And another advantage of this dendrimer is it is having high amount of positive charge, so it can escape from the endosomal degradation. Okay, due to the positive charge, more amount of positive charge. That effect is called as proton sponge effect. So when you add this nanoparticle, you can see here, and when you stain the this one cells with the lysotracker green, or when you add the nanoparticle, you can easily monitor. So wherever the green color is there, we can monitor. We can say that your nanoparticle is attached to the cells and it's entering. And once it is releasing the drug, the red color drug, the epirubicin drug, it is targeting and going to the nucleus. So we can easily monitor where your nanoparticle is and where your drug is released. Okay. So this way it will be easy to monitor the drug release. And this work also highlighted in the Atlas of Science. And the next work, what we did is, instead of using the artificial polymer like dendrimer, we used a protein. Protein is highly biocompatible, so we use a protein-based nanoparticle. So here we have used albumin, bovine serum albumin, BSA. So this protein nanoparticle, the advantage is it is highly biocompatible, and also it has a hydrophilic and hydrophobic packet. So if you see that most of the anti-cancer drugs, these are hydrophobic in nature. If you take paclitaxel or curcumin, okay, the curcumin is healthy, okay. So if we take most of the anti-cancer drug, these are hydrophobic in nature. So we can take those drugs and we can dissolve in the organic solvent and we can pack it inside the protein-based nanoparticle. So if you see this Abraxane, this is the albumin-based protein-based nanoparticle, which is the paclitaxel drug, and this is the first commercially approved by the FDA. This is the FDA-approved first nano-based drug for the breast cancer therapy. If you take the Abraxane, So it is basically based on the NAB technology, that is a nanoparticle albumin bound technology. So those days researchers used Taxol as a drug for the breast cancer. So in the Taxol, for dissolving 6 mg of paclitaxel, we have to use this much amount of organic chemicals, okay, chromophore or ethanol, so which is toxic. So when compared to this, we can, using this nanoparticle based technology, albumin bound technology, we can dissolve 100 mg of paclitaxel using 900 mg of albumin. And here we are not using any surfactant or any solvent. Okay. And you can see the drug here, it is only 6 mg and here it is 100 mg. So we can load 100 mg of paclitaxel in the albumin based nanoparticle, and protein is highly biocompatible. So in this way, we can reduce the toxic effect, and also we can increase the amount of drug. So when you increase the amount of drug, we can reduce the dosage, and it will be having more high therapeutic efficiency. So in our work, what we did is we use the drug repurposing. So drug repurposing means we can use the drug, which is essentially for the other function, and we can use it for the various other applications. For example, here we used niclosamide. So this drug is mainly for killing the tapeworms. Okay, it's mainly for killing the tapeworms, but this drug also have very good anti-cancer effect. Okay, so what we did is, but the one drawback with this drug is this is highly hydrophobic in nature. So we dissolve this drug in the organic solvent and we pack it inside the protein. And this is a very simple method to make the protein nanoparticle. And this drug is entrapped in the protein nanoparticle. And this is how the drug look like. And when you load it on nanoparticle, it will be like a spherical, and the size is in the range of 200 nanometer. When you have a nanoparticle of 200 nanometer, it can be easily go through the passive targeting. And you can see here, when you study the cell viability, and when we add only the drug, when you add only the niclosamide, due to hydrophobic in nature, it is not able to enter the cells and it is not able to kill the cells. But when you put it into the protein nanoparticle, so with respect to the concentration, so it is able to kill the cells. Okay. So in the lung cancer cells, it was IC50 is 5 micromolar, and in the breast cancer, IC50 is 2.6 micromolar. So it is able to induce the cell death. And the next approach is we used nanofibers for the drug delivery. Okay. So you can see here, this is a nanofibers are the continuous fiber, and which is in the range of diameter 50 to 5 nanometer. And when you see this picture, this brown color is the human hair. So the human hair is approximately 80,000 nanometer, and below is your nanofibers. So these are the small size nanofibers, and we use nanofibers for anti-cancer drug delivery. So how to make these nanofibers? So it is similar to making the cotton candy. So you might have seen this cotton candy in the exhibition or during your childhood. So what he do, he add the sugar and he apply the electrical this high voltage power supply to here, and the sugar will melt and it will form the fiber. Okay, so basically what we are eating is a micro size fiber, that is a cotton candy. The same principle here. So in the electrospinning equipment, we can use the electrospinning for making the nanofibers. The principle of electrospinning is similar to the cotton candy. Here we'll be adding the FDA approved polymer, and we'll be adding the any FDA approved anti-cancer drug. So instead of pink color, we'll be adding the anti-cancer drug here. And when you apply the electric voltage, electrostatic potential, due to electrostatic potential, it forms this kind of nanofibers, and these fibers will be collected in the collector. And this is the anti-cancer drug loaded nanofibers. So here we loaded with curcumin. This is a haldi. So that's why this nanofiber mat look like yellow color. So this is the final product. The final product will look like a nanofiber mat. So basically this can be useful for controlled and sustained release of anti-cancer drug, for preventing the local reoccurrence of tumor after the surgery. So when the doctor removes the breast tumor, so what happens is there's a high chance for reoccurrence of the tumor. So to prevent the reoccurrence of the tumor, we can use this kind of nanofiber based bandages, so which can act as a physical barrier, and also it can release the drug slowly and it can induce the apoptosis. Okay, so it will prevent the reoccurrence of tumor. That is a major advantage of this anti-cancer drug loaded nanofibers. So you can see here, this is a video which we taken. So this the small thread-like structure, these are the nanofibers loaded with anti-cancer drug, and you can see here these are the cells. So basically the cells are like elongated, and when it undergo apoptosis, it become rounded. And we can also make a core shell nanofibers. So core and shell nanofiber is similar to your pen and refill. So we can have a core. Okay. So in the core, we can load one type of anti-cancer drug, and in the shell, we can load another type of anti-cancer drug. So that is the advantage. For example, in this work, we have loaded the anti-cancer drug 5-fluorouracil in the core, and in the shell, we loaded curcumin. Okay. So we loaded two types of anti-cancer drug. So when you load two types of anti-cancer drug, so it will have a more amount of anti-cancer effect, like it can have the synergistic therapeutic effect. Okay. And another advantage is we can also control the release of drug by simple cross-linking. So for example, if you see that here the core is also cross-linked and the shell is also cross-linked. So the drug will be released very, very slowly. But here the shell is cross-linked, but the core is not cross-linked. So when the core is not cross-linked, the drug can be released in a faster rate. So we can also control the drug release also by simple cross-linking reaction. That is the advantage. So when you see the core shell nanofibers under the electron microscope, so you can see we can get this kind of structures. And the next important thing is we have to measure the contact angle of the nanofibers. So your membrane should be hydrophilic in nature so that it can easily attach to the your cells and body. Okay. So how we can check it? By simple experiment called contact angle measurement. So here you can put a drop of water on the membrane. When you put a drop of water on the membrane and take a photograph using the contact angle measurement system, and using the software, we can measure the angle. So if the angle is less than 90, that means your material is hydrophilic in nature. And if the angle is more than 90, your material is hydrophobic in nature. And if it is more than 150, it means your material is super hydrophobic in nature. For example, you are having the plastic sheet, and when you put a drop of water on the plastic sheet, so you'll get more amount of contact angle, which is more than 150. That means it's super hydrophobic. And when you put a drop of water on the normal paper, so it will absorb the water. So it will be less than 90. That means it's hydrophilic in nature. So when you made the nanofibers, so only nanofiber, that is the only polymer, it is hydrophilic in nature. When we add the anti-cancer drug, that is the hydrophobic anti-cancer drug, so the slightly hydrophobicity is increased, but still it is less than 90 degree. So it is good for the therapeutic application. And in the next work, we also use the similar kind of core shell nanofibers. So here we loaded with the another gene called suicide gene. Okay, so this gene will make enzyme. Okay, basically suicide gene is a gene which codes for an enzyme. So this enzyme will convert the prodrug into the toxic drug. Okay, so here we use the prodrug called 5-fluorocytosine. So basically the prodrug is not a toxic drug. It's not a drug. It's a prodrug is not toxic in nature. Okay, and we have added this gene called suicide gene, that is a CDUPAT, that is a cytosine deaminase uracil phosphoribosyl transferase. It's a very big enzyme. So what happens is this gene, this will produce the enzyme. So this enzyme will convert your prodrug into drug. Okay, so it will convert the prodrug into the drug. And again, this gene is also having the GFP. So what happens is when you have the nanofiber and when you add it on the cells, when the cells are added, first thing is the shell will degrade and it will release the DNA, and the DNA will be transfected into the cells. So once the cells are transfected, you can easily monitor by the green fluorescent protein expression. And you can stain the nucleus with the Hochest. Okay, so you can stain the nucleus and the nucleus will be blue color. And you can superimpose the image, then you can easily identify, okay, how many cells are transfected, how many cells are transfected, and how many cells are not transfected with the combination of blue-green expression. And another advantage of this the suicide gene therapy is it is having bystander effect. So the bystander effect means once the prodrug is converted into toxic drug, the toxic drug will be going to the nearby cancer cells, and it will also kill the nearby cancer cells. So that effect is called as bystander effect. Even the non-transfected cells in the tumor region will also get killed by this bystander effect. Okay, so that effect is called as bystander effect. And the another advantage is, so if we are having a less amount of transfection, with the less amount of transfection, we can achieve high amount of therapeutic effect. So that is the advantage of this bystander effect. And this work also highlighted in the Nature India. And in the last work, recently what we have done is, so we made a theranostic carbon darts. Instead of making carbon darts separately and therapeutic material separately and combining and make a theranostic nanoparticle, so we made a theranostic carbon darts. So for that, we use this plant leaf. Okay, so this is a periwinkle plant, okay, which you might have seen in the roadside. Okay, and it is one of the important medicinal plant. So Catranthus roseus. Okay, so this plant have a lot of Vinca alkalides. Okay, and these Vinca alkalides have high specificity for the tubulin. Okay, so when you make the carbon dioxide from this, so it is highly specific for the microtubules. It is binding to the microtubules and making the microtubule fragmentation, and also it is making the tubulin to depolymerize. So when it destructs this structure, then cells will undergo the apoptosis. So we can make this carbon dioxide using the simple method called hydrothermal method. That means you are going to take the plant extract, leaf extract, and heat it at the controlled environment for 200 degrees Celsius for 10 hours. And after 10 hours, you will get the fluorescence carbon dioxide. Okay, and this carbon dioxide are highly specific for your tubulin. Okay, and binds to the microtubules and the cytoskeletal fragments, and it will destabilize it and it induces the cell death. So the cell is like a tent. Okay, it is having this kind of support. Okay, so when you remove this support, the cell will fall down, the tent will fall down. Similarly, the cell is having this kind of cytoskeletal fragments and the microtubule. Okay, so when this carbon dioxide bind to these microtubules, and when it start depolymerizing it, then what will happen? It will destabilize. So as I told you, the cell is like a tent. So when you remove the support of the tent will fall down. The same thing happens here. When you disturb the cytoskeletal structure, then the cell will undergo apoptosis. And if you see that these all the pictures are taken using the confocal microscope, and here we are not used any fluorescent dye. So we have whatever the carbon dioxide we made from the plant, the same thing we added here, and this is giving a very beautiful fluorescence, and it is highly binding to the specific for the microtubule and depolymerizing the microtubule and inducing the cell death. Okay, and this work also highlighted in the Nature India. And with this, I'll end my talk. And I thank all my students, and also I thank all the funding agency, including Shastri Indo-Canadian Research Grant, for supporting our research work. And I thank you all for your patience in listening. If you have any questions, I'll be happy to answer. Yes, so there are some questions in the chat box. So yeah, so the theranostic particles specifically to the tumor cells, the first question. So the theranostic particle, as I told, like we can add RGD peptide. Professor Devika, could you mute your mic? It may give an echo sound. Yeah. So we can add a RGD peptide to the carbon dioxide, or we can also add this folic acid, okay, and which can go and specifically bind to the breast cancer cells. And recently we found that, for example, the last work, whatever I explained, so it is binding specifically to the cancer cell, that plant, Vinca alkaloid plant. And we are also still, the mechanism is not known. So we are still working on that to explore the mechanism. So after making the carbon dioxide, also how this plant is able to retain its property for specifically targeting to the microtubules. So that mechanism is not known. So we are still working on that. So if there's any other questions, you can type in the chat box. And so the next question is like how the toxicity of the nanoparticles can be reduced. So as mentioned by Professor Devika also, the toxicity can be reduced and checked by using we can add the polyethylene glycol and other polymers. And also we can use the carbon dioxide. When you use the carbon dioxide, it is the toxicity will be less because it's basically made up of carbon. So by these methods, we can reduce the toxicity. And when you do the cell culture studies and 3D printed tumor model, and when you study the toxicity before we take it to animal models, so in that way we can overcome the challenges with the toxicity. I hope there is no other questions. Then
Can I ask a question?
yeah, sure. Go ahead, please.
You talk about the ROH generation, reactive oxygen species, using you measure it using flow, right? So
Right,
what is
right.
the timeline? What is the timeline?
The timeline depends on the nanoparticles to vary. For example, when you use a silver nanoparticle, it is very quick, within less than 6 hours. So when you use a polymeric nanoparticle with a drug, so it depends on the drug release profile. So, and we monitor it when there is a cell morphology changes, then we take at that particular time point, and we initially did the time dependent study and we optimized it. So in core shell nanofibers, wouldn't be there a further complication for the release of anti-cancer? So in the core shell nanoparticle nanofiber, there won't be any interaction. So because the core and shell will be separated. For example, it is like I told you, like it's like a pen and refill. You can assume that like pen and refill. So in the refill, if you are changing the color, instead of blue color, you can put the red color refill. So it is not going to change the property of the pen. So the same thing here, like you'll be having the shell and the core. So which is not going to intermix. Okay, so we have to make a polymer in such a way. So it will be a kind of like intact core and shell. Okay, and there is no fusing of the drugs. By simple cross-linking reaction, we can control that also. And so what kind of normal cell lines? Yeah, normal cell lines, if we can use, for example, if we're using the lung cancer cell lines, we can use there's a cell line NCL, I forget the name of the NCI. So we can use the normal cell lines also available. Okay. So if you're using breast cancer or lung cancer, you can also get the same kind of normal cell lines, immortal cell lines from the NCCS Pune from India, or you can get it from ATCC. And what type of anti-cancer? Yeah, so once the question is like the core shell, how once the drug is released, both the drugs will active or not? Yeah, both the drugs will be active. Like for example, it is like attacking the person. The two person is attacking a single person. One person from attacking from the front side and other person attacking from the back side. So the one drug will attack the one pathway. The other drug will attack the another pathway. So when you attack the two pathway, the cells will undergo the cell death. So by this, we can also overcome this anti-cancer resistance. When you use a single drug, there's a high chance for getting the anti-cancer resistance. When you use a multiple drug and when you use a low concentration, we can achieve the same therapeutic effect. At the same time, we can also overcome the drug resistant issues. So that is the advantage of this combination therapy. So I guess there is no more questions. Then yeah, we can end the lecture, and now we can move on to the validatory session. Now I would request Dr. Prachi. Hope. Yeah. Yeah, I'll briefly introduce about Dr. Prachi. I welcome, we are honored to have Dr. Prachi Kaul, Director, Shastri Indo-Canadian Institute, for this validatory session. Dr. Prachi Kaul is the Director of Shastri Indo-Canadian Institute for his office in India. She has been associated with the Institute for more than 16 years now, and she is an alumna of IIT Delhi. Dr. Kaul has served to international and national bodies like Plan International and National University of Education Planning and Administration before joining the Shastri Institute. She is a social scientist with keen interest in studying aspirations and social mobility of the masses and its relation to higher education. Dr. Kaul serves on the advisory boards of a couple of academic and research institutes and joint working groups in between India and Canada of different domains. Now I would request Dr. Prachi Kaul to say a few words about the Shastri Indo-Canadian Institute and also various programs available for the students and the faculty colleagues. Thank you. Now it's over to Dr. Prachi.
Thank you, Dr. Gopinath. I'm sorry for the technical glitches that I experienced this morning and couldn't join the inaugural, so apologies again. Sometimes technology did shed very crucial moment, but reconnects again. You know, it's a pros and cons working with technology. So thanks for putting this together, and I'm happy that I could join for the valedictory session. Very good morning and very good evening to those who are joining from Canada. I hope everything is going well with all of you in the time of pandemic. We on this corridor, we have now shared priorities and shared responsibilities in terms of sharing of vaccine from India to Canada, which is I think a very very friendly gesture from our side to Canada, and of course we cherish the presence of Indo-Canadians in terms of the diaspora's presence there in Canada. So amazing to see things happening on this binational corridor, and I would say that extremely proud to be part of this particular conference, which is a workshop, which is on nano-bioengineering. This is a part of the larger project that Shastri has funded to IIT Roorkee and University of Victoria under the Shastri Institutional Collaborative Research Grant, which is primarily to support and encourage, institutional collaboration in research and development for for the period of 2 years between institutions in Canada and India, in which institutions and researchers can gain from each other and develop their research areas. This grant is directly aims at strengthening the binational research initiatives and demonstrate the collective and added value in working together. The grant have been envisaged. I mean, this is a grant that we have been offering for last 10 years. And I would say this intends to fill, and we have proven record of this, that this has actually intend to fill information gaps for policymakers and others, those who are influencing the policy making processes and issues wherein such researchers are very, very productive are the areas that this particular project is undertaken. This particular grant is proposed on a binational research initiative to support, of course, not just binational, but research that affects the whole community at the world level. And I think this program over the period of time has given and provided a chance to demonstrate the ability to put across innovative ideas at the grassroots level and, of course, at the most scientific and technical oriented researches. One of this is I'm happy to be present in this conference, in this workshop, which deals with the similar ideas and thoughts. I'm sure the deliberations of the event or the workshop went off really well, and discussed the transformations occurring in this particular area worldwide, also under this particular collaboration that is happening with Dr. Gopinath and Dr. Devika ji. I'm sure that this will work as a conduit to build up the capacity of the people involved in the process, especially the younger lot of researchers and students, which are on the priority of workings of Shastri Institute. A large number of these projects that we have granted were really, really impactful in terms of their adoption by the governments, stakeholders, and other organizations for the greater good of the communities in the respective countries of implementation. And I'm happy to share that for the next 5 years to come, we are going to adapt to research clusters, and which are the clusters are going to be based on One Health, ocean and marine life, big data analysis, also artificial intelligence and its uses. I'm pleased to share that, in the last couple of days, I have been invited to be present in the inaugurals of the Shastri funded programs conferences. And I think similar to this event, similar to this project, we have supported almost 10 conferences. And these, very pleased to share that last 10 days only, I have attended at least 6 to 7 events of this nature, wherein we are working towards building up technology that sustains and it helps in cancer care and other aspects of bioengineering too. So, and other aspect that relates to sustainable development and growth of human beings. So, and also about the flora fauna. So I would say that I'm happy that Shastri Institute has contributed immensely in this area and will continue for sure. And as I said, that these research clusters are going to give us very, very focused approach to work on and resolving a problem that remains on this binational corridor and of course worldwide. All the very best for you to explore the opportunities through Shastri, to be more focused in the areas that you would like to continue your association with, either with on this ongoing collaboration or forming a new collaboration with other institutions in India and Canada. This particular event is happening on binational corridor, and I would say these are two great democracies of the world, and we have shared a responsibility to deliver to the world, I would say. And this is very, very important for me to place here that, in very less time, I think our initiation in science and technology and engineering started in 2004-5. And in this time, we have spread our workings in all domains of knowledge related to STEM. Before that, Shastri was only concentrated on humanities and social sciences and development, which remains as a focus area, but we are working now on all domain of knowledge wherever and everywhere possible. We are a binational organization, those who are not aware, and we have a unique mandate to operate in two directions. First, we serve as a financial support system for academic and research programs, like this grant that we have granted to IIT and University of Victoria to carry out research, and similar programs for students. I'm pleased to share that 70% of the funds that we get is spent on students. So we are very, very student-centric organization. When students are funded, it involves faculty, it involves researchers of different capacities and capabilities as well. So it's a comprehensive approach that we adapt, but core of the approach lies with the students. So that is our focus. And secondly, we provide a binational network of institutions with whom you can collaborate. And we consider these institutions as a knowledge partner, which provide you an opportunity to discuss, deliberate, and understand each other, and come together to I would say, solve the problem that we all are facing. So congratulations once again, Dr. Gopinath and Dr. Devika ji, for putting this together. I'm sure everybody has learned a lot and enriched themselves in this particular domain of knowledge. And congratulations to Dr. Chaturvedi as well, the director of IIT Roorkee, for supporting the initiative funded by Shastri Institute. All the very best. Keep on doing this wonderful work, and I wish you the very best. Thank you so much, and thanks for having me here.
Thank you, Professor Prachikol, for wonderful words and supporting us. And also, thank you, Dr. Reshma Rana, for joining this validity session. And with this note, this one-day workshop is concluding now. And all the participants will be emailing you the certificates to the registered email ID. We'll be sending the certificates within a week time. Okay. So I also thank all the participants and all the committee members who helped in organizing this one-day workshop. And thank you all. With this note, we'll end this session. Thank you.